Professor, faculty members, teachers, and dear students. I, Tabassum Jahan, on behalf of the organizing committee, welcome you all to this 3 days international conference cum workshop on proteomics, transcriptomics, and molecular drug design and integrated omics approach. This omics is not a new science as it dates its origin back to the late 90s with the human genome sequencing. It ushered an era of genomics. Following that, with advancement in technologies, in statistical softwares and databases, it graduated to and diversified to various fields like proteomics, transcriptomics, metabolomics, and so on. We felt the need of keeping ourselves as well as our students abreast with the basics as well as the advancement that is taking place in this field. So this events, it has talks from the experts as well as it aims to provide some hands-on experiences also with our hands-on sessions, which aim to give an exposure of how we actually perform the experiments and the studies in the lab. Now, with these words, I would like to request our principal, Sir, Professor Narendra Singh, to address the delegates. Over to you, sir.
Namaskar and good morning to the participants of this conference. And it gives me immense pleasure in welcoming participants to this international conference and workshop organized by Department of Botany. The theme of the conference and the associated workshop is of great significance, and I am confident that all the teachers, researchers, and students would be greatly benefited by it. As I understand, the topic of the conference is extremely relevant in the present day context with tremendous applications in agriculture, disease management, drug discovery, and drug designing, and also in basic research. The conference will give good exposure to the participants in terms of technique as well as applications. The conference is also associated with hands-on experiments. It is a good idea since it brings forth the challenges of technique. Basically, when you perform experiments, you learn so many things. So it enhances your understanding. Department of Botany has always been active in organizing such conferences, workshops to educate their students and keep their faculty members informed about the new developments in their respective fields. I congratulate the department and wish fruitful deliberations for next 3 days. And once again, I welcome all the participants on the behalf of college and the organizing committee. Thank you. All the best.
Thank you so much, sir, for your kind words of encouragement to us. Now, we would like to release the abstract book of the event before this August gathering. Can I request Dr. Savinder to kindly present?
Okay, please let me know once my screen is shared.
Yeah.
Is it visible, ma'am?
Yes, it is visible.
Okay, thank you, ma'am. So I'm sharing the first few pages of our abstract book, and this is the cover. Followed by the brief introduction about our distinguished speakers. And these are our resource person for our hands-on sessions. Our advisory board. And this is our team. This is about our college and Department of Botany, University of Delhi. So it will be followed by the message forwarded by principal sir and message from head of the department. Then we have given a brief CV of our speakers and resource person. And then it will be content. And after that, you will get all the abstracts. I think this is sufficient for now. Is it okay, ma'am?
Thank you so much, Dr. Savindra.
Okay, ma'am. Thank you.
And I would like to mention here that post-conference full-length paper will be invited from the abstracts, and we have tied up with an international peer-reviewed journal for publishing those papers in a special issue. Now, I would request Dr. Ratnam Kaul Vatal to kindly take over and introduce our speaker for the inaugural talk, Professor Sandeep Das. Over to you, ma'am.
Thank you, Dr. Tabassum. Good morning and a very warm welcome to all assembled on the online platform. It gives me immense pleasure in welcoming our own illuminous Professor Sandeep Das to the inaugural session of the International Conference cum workshop today. Welcome, Professor Das. I would like to introduce Professor Das for the participants. Dr. Sandeep Das is a professor in the Department of Botany, University of Delhi. As I've mentioned, he is an alumnus of 1987-90 batch of BSc Honours Botany of Zakir Hussain Delhi College. He did his PhD from Jamia Hamdard and Delhi, and he has a long postdoctoral research experience from Cornell University, USA, and Max Planck Institute of Developmental Biology, Germany. His major research areas have been, His major research areas have been genomic studies in members of the family Brassicaceae, evolution of plant morphological diversity and adaptive variation, role of microRNA and transcription factor genes and cis-regulatory elements, impact of polyploidization on regulatory genes, genetic networks, and regulatory diversification and genome organization of plants' molecular and functional diversity. He has guided a large number of MPhil, PhD, and postdoctoral researchers. He has 22 years of teaching and research experience. Professor Das has several research publications to his credits in journals of repute, and he is a life member of several academic bodies. With this brief introduction, I welcome Professor Das for the inaugural session of this international conference. Over to you, Professor Das.
Good morning to all of you. Am I audible clearly?
Yes, yes.
And thank you, ma'am, for the generous and very kind invitation. We are still, I think, your students of the college, and I'm extremely grateful, that the college has given us what we are today. But before I begin my talk, let me just share my slides. So please let me know if my slides are visible to all.
Yes, sir, it's visible.
Okay, just a second. And is it in the slideshow mode?
Yeah, it's in slideshow mode, sir.
Okay, so this is just a snapshot to show that the dawn is here. And I hope that we'll have better times than before. Okay, so again, before I begin, let me thank the organizers. I think Dr. Tabassum and Dr. Ruchi contacted us, asked me, and I was, uh, overwhelmed when I got to know that Zakir Hussain is organizing this. And I just highlighted that, three institutes with a great legacy, 300 years plus legacy or heritage of Zakir Hussain College, University of Delhi, which is 100 years, and the Department of Botany, which is completing 75 years. So I think it could not have been a better time than now to triangulate the three, the college, the department, and the university on the same platform. And thank you once again for the kind invitation. And I think as Professor Ratnam mentioned, I dedicate this talk to my teachers and staff of the Department of Botany. I think I remember Professor Virendra Kumar, Dr. Harjinder Grover, Dr. Vishwas, Dr. Sunita Puri or Aisha ma'am, Dr. Vishwas, Dr. Mrinal Bhargav, and others. I think in the staff, I still remember Pritpal ji, Afsar, and other staff who were there when we were students of BSc in 1987 to 99 and to 1990. And then I also was registered for MSc from Zakir Hussain College itself, 1990 to 1992. And we used to regularly visit the college for library work and others. So my basic training, my journey began with what we achieved from this college. Okay. So before I begin this talk, I believe that there will be a lot of students who are participating. So what I did is in my talk, I have basically eliminated a lot of technical details and kept it relatively simple for me to explain to the audience. But again, if anybody has any questions or any clarification, you can stop me in between and I will try to address that. Okay. On and so forth. So the work that I presented, they were published. One is a work of an MPhil student, which was published in 2016. The other is a part of a PhD student who is still working and going to submit her thesis now. Now, this organization, what does it lead to? As I mentioned, does it have any functional? I mean, this cannot be just an academic exercise by themselves. So is it important to study the function? And lastly, I thank my research scholars. I mean, although I presented, but it is all the hard work of the students, postdocs, trainees, JRFs who have worked here. Today, I presented some work which was done by Saurav for his PhD, Priyanka for her MPhil, Dipali, who was a JRF who left last year, and then some work from Ekta and Mukund, who have done the work on sulfate and others, and the meiosis, stamen, anther, Richa Shukla, Ekta Pokhriyal, Komal Jalan, Shubhangi Pandey, and Kavita Devi. And thank you for patient hearing. I would be happy to take questions, comments, criticisms, if any, and should be there. Thank you so much.
Thank you, Professor Das, for a wonderful lecture. Honestly, I'll take a moment to take pride in the fact that you are a Zakarian, and this should be an inspiration for our present day batches, especially, at the moment we have, most of them are from first year, semester 2, because second year and third year are in their final semester and exams. So first years, the ease with which you have explained, the simplicity of the explanation, it was wonderful. I really enjoyed, and a person like me who's definitely not an omics person, I really could get quite a lot, not 100%, but quite a lot of information. I also felt that the decision to, I noticed in your CV that you have continuously worked on members of Brassicaceae, and I think it was very creditable because you had two-sided sword to tackle the challenge. One, it was a Arabidopsis model plant, and the second important crop plant. So that was it. Now, there are a lot of questions. I really don't know how many we'll be able to take, but let's start somewhere.
Can I just add one? Can I just add one? I think the credit for Brassicaceae I must give to Professor Shivanna. Because he is the one who introduced us. I think a lot of us from Department of Botany were introduced to Brassica by Professor Shivanna, and I think I must give this credit to him.
Yes, Professor Shivanna was a great professor of botany, and he still continues to contribute to the field. So my first question, of course, not my question, from the participants, can change in non-coding region alter the health or behavior of organisms?
Can you, ma'am, please?
Can change in non-coding region alter the health or behavior of organism? Any genetic change in the...
Yes. So, for example, non-coding RNAs are known to be, what we call as master regulators. So there are many examples. I mean, I can think of plants, I can think of animals, humans, and certainly I cannot think of behavior because that's a very complex subject. But yes, health now depends on what we call as health. I mean, in terms of, let's say, development. So for instance, if I think of, let's say, plants, so for instance, vernalization is regulated by, so if I think of plant development, reproductive success, so vernalization is dependent on small RNA mediated regulation. So that's one small example. There are many small RNAs or non-coding RNAs that are involved in biotic stress responses and abiotic stress responses in plants. So certainly it is in plants. In human health, yes, many, for example, in many cancers, the regions that are most prone to chromosomal reorganization are known to be involved in production of small RNA. So yes, small RNAs. I remember there was a study when loss of calcium, from bones was linked to small RNA. So certainly it is a major regulator and of both human and plant health.
Okay, thank you. The second question is, can the promoter regions used for making universal primers, which can work across various taxa?
It's a very good question, but again, you see with promoter, the problem is promoters, there is no defined, like in genes. Why genes are easy to study is because genes have a very defined boundary. You know that a coding region will begin with ATG and end with whatever stop codon it is there, translational stop codon. With promoter, there is no defined boundary except that we think promoters are between 1 kb, 1.5 kb in length. That's one thing. Second thing is promoters tend to evolve more rapidly than coding regions. So there is what is called as regulatory diversification within promoters across different species. So in many times, and that's what we see even within, Arabidopsis brassica, the way Arabidopsis promoters work is not the same way Brassica promoters work because there is a lot of sequence diversity. Now, I think I don't know who asked that question, but it's a very good question because translation of information, whether for markers, whether for characterization, can occur if there is some sequence homology, functional homology, and so on and so forth. So promoters, I would not be very comfortable in saying that yes, the promoter of Arabidopsis will be the same as promoter of Brassica. No. So unless we prove it experimentally, it's hard to say. With proteins, with genes, it is slightly easier to say.
Thank you once again. Are microRNA present in all plants?
Yes, they are universal. They are universal.
Microorganisms also?
So in the bacteria and viruses, the whole CRISPR technology is based on small RNA, but there we do not call it microRNA. It's given a different name. But essentially the process, if you see, there is also a guide RNA, here is a microRNA. So I believe they would.
Matter of terminology, you would say?
Plus, I think microRNAs and small RNAs evolutionary arose because of as a defense response. When viruses attack plants, so the plant produces small RNAs to silence the viral genome through methylation and other things. So viruses, I would not say, but bacteria, there are small RNAs present. Yes.
Can we transfer? Sorry, yeah. My question was, can we transfer microRNA from one to the other plant? Is there a technology?
Yes, you can generate transgenic lines, because what happens in microRNA is the 21 base pair region is the one that is most critical in function. So what we use, for example, is a technology called as an artificial microRNA. So not all genes are regulated by microRNA. We can use what are called as artificial microRNA. So based on the principle, actually now we can design microRNA for anything we want, based if there are some criteria that are met. So yes, we can transfer from one to the other. Yeah, that's not a problem. We do it.
So they can be used as biomarkers then? No.
But if you're transferring, then they cannot be used as biomarkers. Have to come from the endogenous region itself. So for example, if I'm transferring one microRNA from, let us say, rice to wheat, so it cannot be a biomarker for wheat. For a wheat biomarker,
for rice it would be, yeah.
Rice it would be, yes.
Yeah. There's a question. What is the meaning of non-canonical organization?
So it's a very general. So canonical means what is commonly observed. Non-canonical is what is very rare, what is unique organization. So non-canonical is not a very common phenomena. We don't see it very common.
Last two questions. How gene silencing plays a vital role in omics research?
So today, I think the entire field of omic research is more or less centered around generating mutants, a lot of mutants, and many of them are based on using gene silencing technology. So I would not say it is the central pillar, but it is one of the most important, because as I mentioned, so artificial microRNAs we can use to silence, which then can be used for omics research. So for example, I did not show the last part where we talked about that stamen example. So it is a mutant which is based on gene silencing that we have generated in the lab. And then we have done transcriptomics of that to know what changes it is making. And I think there are hundreds of such examples. So it is something, I'm saying that this is not something that we have done new. It's a very commonly used strategy.
Thank you. And one of the participants has asked, what is GFP and Gus?
Oh, so
they have not started yet.
No problem. So GFP is a green fluorescent protein from a fish, which is a fluorescent. It gives a fluorescent green color when you excite with, I think 495 nanometer wavelength. And Gus is the beta urodinase gene from E. coli, which produces an enzyme which cleaves X-gluc as a substrate or umbelliferon as a substrate to produce indigo colored product on oxidation. So GFP, the gene for GFP can or has been mutated now through site-directed mutagenesis, through protein engineering, and now we have cyan fluorescence color, we have yellow fluorescent color, we have red fluorescent color. So because then we can use multiple fluorochromes for simultaneous detection. So I hope that answers the questions.
Yeah, I'm sure. Thank you so much, Professor Das, for a very enlightening session. And I wish we can call you offline and have a session.
Actually, I must. Yeah, so I must. I was little when Dr. Ruchi said it's going to be online. I was little. I said to you, I mean, when I initially said yes, I thought that I could go offline, meet my teachers, and be in the college, and I'll be very, very happy to come to college any day. I mean, now my exams are over. I'm only doing some thesis. Whenever, ma'am, you want, I will come to the college.
We'll definitely organize an offline lecture. This had to be online because you see, we are poor people and we couldn't organize offline international conference, which we really wanted to do. So that is why. And thank you once again on behalf of the department, on behalf of college, and on my personal behalf.
Thank you,
Thank you.
Thank you so much. And thank you to all the organizers who are here for this opportunity. Thank you, ma'am.
Welcome. Now we would go over to the hands-on session, and our speaker for the hands-on session is Ms. Diksha Bisht. So let me introduce Ms. Diksha Bisht, who's a PhD scholar since 2019 at Department of Plant Molecular Biology, South Campus, University of Delhi. And she's pursuing her PhD under the able guidance of Professor Girdhar Kumar Pandey. She completed her bachelor in botany from Shivaji College, University of Delhi in 2016, and master's in plant sciences from Central University of Punjab in 2018. During her master's, she worked on differential alternative splicing analysis under abiotic stress conditions in barley, using Galaxy Web Server as MSc dissertation with Dr. Pankaj Bhardwaj. After that, she worked on oxidative stress condition in IARI, New Delhi, as a young professional, YP2, it's a placement. Currently, her PhD work involves various calcium sensors and kinases regulating the calcium signaling network in plants under different abiotic stress conditions. Her work mainly focuses on the genomics and transcriptomics study of kinases under nutrient deficiency conditions to understand the mechanistic pathway of calcium signaling calcium signaling cascade. So I welcome Dr. Diksha, Ms. Diksha, and over to you, Diksha.
I would request Dr. Ruchi to please play the hands-on session of Ms. Diksha Bisht. And she is here to clarify any doubts or any questions that arise.
Yes, ma'am, and thank you for the very good introduction. Thanks a lot. Hi everyone, this is Diksha Bish and I am the PhD student in Department of Plant Molecular Biology.
Is my screen is visible? And was the video audible to all of you?
No, video is not playing. Screen is visible.
Okay, ma'am. Just
Hi everyone, this is
is it
Diksha
visible
Bish
and
I am
audible
the
now?
PhD student in Department of
Sound
Plant
is
Molecular
coming, but
Biology,
I can't
University
see
of
the video.
Delhi. So today I am
Just a
going to tell
moment,
you
please.
about the RNA seq analysis and the data interpretation using the web interface.
Now is it visible and audible?
No, Dr. Ruchi.
I would request if Ms. Diksha has this video, can you play that? Can you share your screen and play that video?
Yes, ma'am, sure. Okay, I think it's visible now.
Yeah, it's
Ruchi ma'am.
Is it visible and audible now?
yeah, it is visible but audible. We have to see.
Just check, Dr. Tabassum. Can you hear the sound?
No, Dr. Ruchi, sound is not there. When you are sharing, please check the computer sound on your right.
Is it audible and visible now, Dr. Tabassum?
It's visible, but the sound is yet to come. I mean, we can't listen anything.
Analysis using the Galaxy web server. Here you do. Hi everyone, this is Diksha
Is it
Pesh.
Yeah,
visible?
I'm the PhD
yes,
student
now
in
it is.
Department of Plant Molecular
Thanks,
Biology,
Dr. Ruchi.
University of Delhi. So
I
today
am
I'm going to
playing
tell
it
you
from the
about the
beginning.
RNA Seq analysis and the data interpretation.
Okay, ma'am.
Hi everyone, this is Diksha Pesh and I am the PhD student in Department of Plant Molecular Biology, University of Delhi. So today I am going to tell you about the RNA seq analysis and the data interpretation using the web interface. So the web interface which we are going to use today would be the Galaxy web server. Okay, so here we will see like how we can do the NGS data analysis using the Galaxy web server. Here you do not need any kind of programming here or any kind of bioinformatic expertise. So let's start with the hands-on workshop for the RNA-seq analysis using the Galaxy web server. So first, let me give you a basic introduction to the NGS dataset generation and the quality scores. So what is NGS? NGS is the next generation sequencing data, and it refers to the collection of technologies which utilizes the massive parallel sequencing approaches to produce a millions of short reads in a much shorter time as compared to the traditional methods of sequencing, that is single sequencing, right? So it is exploited to determine the order of nucleotide or the sequences of the nucleic acid or the specific target regions of the DNA and RNA. So basically there are many platforms, NGS platforms are available, but the Illumina is the most popular among the currently available NGS platforms because it offers the various of scalable options which complements the requirement of our different study designs and the cost of sequences and also the application of the sequencing data, right? So Illumina sequencing method or the NGS platform Its data analysis includes the three main steps. First is the primary, secondary, and the tertiary. So the primary is the real-time analysis. It basically provides the base call and the associated quality scores representing the primary structure and the primary structure of the DNA and RNA helices. Right. The secondary structure involves the alignment in the assembly of the nucleic acid fragments and provide the complete sequence for the sample. Okay, and the tertiary step, it involves the biological data mining and its interpretation too. So now let us look towards how the workflow of the Illumina works, right? How the Illumina sequencing method or the strategy works. So here is the diagram for that. First, let me erase the previous marks which I have done here. It is coming in between of diagram. Right. Okay, let's start with the workflow of the Illumina sequencing. So what happened here is, first the DNA, genomic DNA is fragmented into small size fragments, right? And then the adapters are ligated to create a sequencing library. Okay, then these reads with the adapters are subjected to the flow cell. Okay, then then these fragments are hybridized to the flow cell surface, and each bounded fragments it is amplified. Okay, and they are amplified to form a clone of itself. So these are known as clone clusters. Okay, these are produced through the amplification. Now these clone clusters are subjected to the sequences by synthesis with the reversible terminator. So here what happened is each of the nucleotide ATGC, right? ATGC are labeled with the specific fluorophores. There are different colors which are emitted when the specific nucleotide is incorporated into the nucleic acid chain, right? So the nucleotide are labeled with the specific fluorophores. These are will be added at the 3' end of the primer, right? These are added at the 3' of the primer and is added at the 3' end of the primer. Then the fluorophore signal will be emitted by the fluorophore uh, will be emitted by the fluorophore when it get detected by the detector. Okay, right? And then the 3' cap will be cleaved and it allows the next cycle to begin again. So that is how the different that is how the next cycle is getting repeated as we are using the reversible terminators here, right? So that is how the next chain sequencing of the Illumina work. So now we are going to focus on the advances in the sequencing technologies, which involves the paired end sequencing and the single end sequencing, right? Earlier, the single end sequencing was done. So in the single end sequences, it involves the sequencing from only one end or the in the one direction, right? And in the paired end sequencing, it involves the sequencing from both the ends of the DNA fragments in the library. So basically what is helped is these read pairs from both the ends, right? And from both the ends, it enables a more accuracy of the read alignment and its ability to detect the indels, right? The insertion deletion variants, which is not possible with the single end data, right? Also at the same time it facilitate the detection of the genomic rearrangement and the repetitive sequence elements as well as the gene fusion or the novel transcript of the gene, right? So the paired end sequencing is always better because it involves the sequencing from both the end of the fragments. Okay, now if I talk about the Fred quality score, so it is important to know the Fred quality score of your data because it assesses the base call quality of the base call, whether the bases which are incorporated ATGC which are incorporated while the sequencing of your data, whether it is how what is the quality of for that involvement or the incorporation of that particular base in the at the particular 3' end. So okay, so this is generated by the Fred program scan. It creates the chromatogram or the peaks and on the basis of that it provides the scores. Okay, so it discusses the certainty or the accuracy of that particular base which has been incorporated into your sequence. So the Fred quality scores, it is basically uh, it is basically available in the 10, 20, 30, 40, 50 and 60. So here the 10 means that one base call is incorrect in when we are talking about the 10 bases, right? uh, from the 10 bases only one base is incorrect, right? When the Fred quality score is 20, then it means the 1 in 100, like in 100 bases the one is incorrect, right? When the quality score is 30, this means that one is incorrect in 1000 bases incorporated. And when the 40 is a Fred quality score, then it means that in one 10 in the 10,000 incorporated bases only there is one incorrect base call is there. So as we are increasing the Fred quality score, the base call accuracy is increasing, right? So what does it mean? What does it what it is showing? It is showing that the Fred quality score, it determines the quality of the base call, and as well as when the Fred quality score is increasing, the accuracy, your accuracy of the base calling is increasing. So it means that if you are going to talk about the quality of your data set or the NGS sequencing data, and so you will look towards the Fred quality score. And if you see the Fred quality score is of 60, right? If your sequences has a 60 Fred quality score, so this means the accuracy of your sequencing data is 99. For that basis would be the 99.99%. Okay, so always we prefer the Fred quality score should be above 20 or after 20. So you can say that the quality of your sequencing data is a very good, right? Now let's talk about the output file of the sequencing data, right? So in the sequencing data of the Illumina, the output file will be the Fastq file. So how the Fastq file looks like? So Fastq file looks like somewhere like this. Once we have a header here in the sequence, nucleotide sequence, and there is a separator which is denoted with the plus sign or a plus symbol, and then there are the quality score, right? In the header there will be the SRA ID, SRA database ID, and then there is a sequence, right? The nucleotide sequence and separator with the plus sign. Okay, now we will look for whether what are these quality scores and how these quality scores are encoded or generated. So the quality score encoding of the Fastq files. So basically in Fastq files format, these quality scores are encoded in the compact form, which uses only 1 byte per value. So to decrease the size of the file, they have encoded the quality scores of each of the bases incorporated, right? So in this encoding, they have used the SI2, or we can see the SI code, which means here you can see that in the Illumina platform, in Illumina 1.8 platform, it uses the SI code means the Fred quality score plus 33. Okay, it is adding with the score, it is adding the 33 value. So if here these symbols are all these symbols have some value according to the coding. Okay, so this means this exclamatory symbol has the 33 value, plus 33 with the Fred score. Plus 33 means the Fred score would be 0 here. 0 means it has a very low quality base call, or the Fred score is 0 means the base is incorporated is of very low quality, right? Here the quality score is 59, right? And it is denoted by the colon symbol. Colon symbol is 59, so the Fred quality is plus 33. So it means that the 26 is the Fred quality score. So you can say that it is still better because it is more than 20. So the Fred quality score will be 1 in 100. So means in 100 incorporated base, there is only one is incorrect, right? So that's how we do the encoding of the Fastq files, and we provide the quality scores to the Fastq file. Okay, all right. Okay, so till now I have discussed about the NGS dataset generation, the Fred quality score, and the single-ended and the paired-ended sequencing. So now I will be discussing about the Galaxy web interface, the main hero of the hands-on workshop. So Galaxy is an open source web-based platform which is used for the data-intensive biomedical research and also to analyze the large dataset which are found in the genomics, transcriptomics, proteomics, metabolomics, and imaging. So today here we are going to focus on the transcriptomics data. Now these are the website which is the usegalaxy.org that can be used to access the Galaxy web interface, and for the plant users or the researcher which are working on focusing on plant, so they can use the plants.usegalaxy.eu. So EU here stand for the Europe. There are the 3 web servers available for the Galaxy. One is from the region of Europe, another is from Australia, and from US. Okay, so what Galaxy makes? Galaxy makes the bioinformatics analysis accessible to the users which do not have the programming information or the knowledge, or who lacks the bioinformatic expertise. So they can use easily or access the workflow here in the Galaxy through a point and click interface, right? So there are some prerequisites for the Galaxy using the Galaxy interface and the NGS data analysis on Galaxy. The one is the computer which supports the JavaScript and the HTML5, and also the Internet Common Web Browser, that is Firefox, Chrome, Safari, Opera. One can use any one of these browsers, right? So now for NGS analysis, the prerequisite are the web browser which support again the JavaScript and HTML5, and the main important thing is access to the internet, the good speed internet, and the user account at the Galaxy public public Galaxy server or any local Galaxy installation which they can use using the cloud, and the NGS dataset obviously in the Fastq format. The Fastq format is important for analyzing the data in the Galaxy, right? So here, if I talk about the basic terms and the descriptions which we will be using for in the whole process of using the analysis NGS dataset analysis. One is the data set, right? The data which we are going to the input and the output from each of the step in the analysis. Another is a tool there are various tools which we are going to operate within the Galaxy, and then there is a history panel is there in the Galaxy interface, and then there is a workflow or the pipeline which in the RNA Seq or in the bioinformatics, the workflow we will call the workflow, like if we are doing anything and there's a different steps in that, then we call it as a pipeline or a workflow, which allows the number of analysis step to be performed automatically, right? So and then there is an instance that like the Galaxy instance in a single occurrence of the Galaxy server. Like there are the different Galaxy servers that can be run separately, okay, at any particular time. And then the main Galaxy public instance, that is I have already told you about the website, that is usegalaxy.org. Okay, so now this is the Galaxy analysis interface. Once you log in the Galaxy website, the website which I have already told you, the usegalaxy.org, then this page will open, this server will open, and from this user here, you can create or register your ID and log into the Galaxy web server using that particular ID and password. So here in this interface, you can find the main 3 important things. One is tool menu at the left, another is history panel at the right, and the tool interface in the middle or the center of the server. Okay, so in the left tool menu, that is denoted with the B, you can find out the different tools which can be utilized, or the software which can be utilized for their data analysis, right? Here the different tools for the NGS data analysis or the software will be present. So these tools are organized into sections based upon the functions, and when you click on the particular tool, these tools will appear on the middle panel, and in the middle panel you can select your input data set to configure the tool parameter setting, and then you can press on the execute button to start your analysis, and Once you press on the execute button, then the result of your analysis will go to the history panel. So this history panel will contain the output data set from every step performed during the particular analysis, right? Once you perform analysis in the first data set, then it will come as output in the here in the history panel, right? So here in the history panel, as you can see, one is labeled as G, E, H, F, and D. Okay, so D is the D belongs to the history panel, and what this G shows, this is the green colored area is over here, right? So this green color tool shows that this file has been successfully executed, and this data set is ready to use. You can use this output data, right? In the yellow color, once you upload any of the data, then it will come in the yellow color, that it means it is still under process and it is getting executed. And when there is gray color is shown, gray color means that this data you have just uploaded, and when after the gray color it will become yellow color, it means that the data is under process, right? And whenever you see that the output or the data set it is in red color, so this means that there is some error has happened during the analysis step, or there is some error with this data set. Okay, so this green color means that the data set, the output is good and it ready to be used. Okay, so now this is the RNA Seq analysis workflow which we are going to use with Galaxy, right? So first we will have the raw data from the Sra database that is available from NCBI, the publicly available data, or you can use your data from RNA, right? RNA sequencing, any of your data, and then followed by the quality control check and trimming that will be done by the software using Fastq and Trimomatic, and then alignment to the reference genome with the help of HiSet2 software, and then the counting that will generate the count file using the HTC count, and then the identification of the DEGs, differentially expressed genes, and later differentially expressed genes, and later on the extraction and the annotation of those genes using the annotate DSeq2 tool in the Galaxy. So all of these things we are going to perform in the Galaxy, where you do not need any coding information or the prior R in how to use R. You do not know the information of R or any programming language. You just need to do like how these things or the tool can be used in the, you just need to understand like how these the workflow and the pipeline, and how these tools can be used in the Galaxy web interface, and how to perform with the raw data. Okay, so let's start with the extraction of the NGS datasets from the Sra database. So Sra database is the sequence read archive. It is a database of NCBI which stores all the raw sequencing data of the alignment information. Uh, it enhance the reproducibility and the facilitate the new discoveries through the data analysis, right? So in this Sra database, you can search over here in this panel. You can write for the data you want to search. Like if I want to search the data about the Arabidopsis data or the plant data sets under different stress conditions, so that here I can write uh, Arabidopsis under different abiotic stress condition. So the list of the conditions will appear here, and I can from that conditions I can look into the particular experiment which I want to analyze, or I want to do it further. Then I can select those data. Okay, so here for this workshop I have selected the data from this accession number or the Sra ID, you can say. uh, Here the data contains, in this ID the data contains the wild type, that is the control, and another is the salt stress condition data of the plant sample Arabidopsis. Salt stress, or we are this is the treatment, And here this wild type contains the three replicates, that is R1, R2 and R3. And here the treatment also contains the three replicates R2 and R3. Okay, so in these two between, in this small amount of data set I have chosen for this hands-on workshop. So here you have to find out the DGs, differentially expressed genes, that are present in the treatment condition as compared to the control condition under the salt stress. Okay, so here once we figure out for the data which we want to analyze, we will search over here, and then this page will open. Here this page will give you all the necessary information about this data. Once you will get the ID here of the SRA ID, and what this data is about, the RNA seq of col0. Col0 is the file type, ecotype of Arabidopsis. So this data belongs to the seedling, and it will tell you about the Illumina HiSeq, the sequencing instrument which was used, and the number of runs, and here the size of the file, and the amount and the number of bases. Okay, so this abstract will tell you about the experiment which was done for this generation of this data set, and here you will get all the information like this is the Arabidopsis thaliana, this mutant modulated transcriptome under the biotic and abiotic stress conditions. So from this we are using only the abiotic stress condition data. So once you press or click on this all experiment over here, then you will get the different page. Then there is a new page will open, and that page will look like something like that. Here all the different replicates of these experiments will come up, right? And all and from these you can get the SRA ID of that particular data. From these run ID which I have got from the SRA database, I am utilizing these run IDs, these SRR IDs in the another database, that is ENA, European Nucleotide Database. In this European nucleotide database, from here you will get all the sequencing information covering the raw sequencing data and the sequence assembly information and the functional annotation, right? From here, from the ENA, I can download the Fastq files, that too in the zip format. Okay, so from here what will you do is, once you search your run ID, SRA run ID from in the ENA database, then this page will appear. This page will give you the same information about the sample accession, the sequence platform of the Illumina, okay, and the model and the number of total read counts, okay, and the number of total base count, and then it will tell you about whether this data is paired ended or the single ended, and the library is for the RNA seq library, and source is the transcriptome, right? From here you have to tick on both of these Fastq files, because it is the paired ended data. You will get two reads, read 1 and read 2, for each of the file. Like I have three file types, three replicates of file type, and three replicates of treated condition. Okay, T1, T2, 3, T3, and C1, that is control, C2, C3. And for each of the three condition, we will have two reads, R1 and R2, R1 and R2. Okay, so we have to download all the two reads for each of the file, like for wild type also and for treatment also. Okay, so once here it is written that once the experiment will open, all the information about the sample, you have to search with the SRA ID or the run ID, and then select both the Fastq files for each of the wild type and each of the treatment condition, and then it will be downloaded in your system as the zip file, and then you have to extract those files from the zip file. Okay, so now once you have downloaded the data in your system and you have extracted the Fastq files in your particular folder, okay, now what you have to do is to upload that Fastq file in the Galaxy. So here what I have used is use use plantgalaxy.org because I am using a plant dataset. Okay, so here what I have done is first I have to create a new history here. With this plus sign, I'll create the new history here, and then this history we can rename by clicking on here. In the history we can rename our history, and I have renamed it to the today's workshop session. And then what you have to do is you have to click on this upload data, and from this upload data this page will appear, this panel will appear, and from here you go to the choose from local files, and then when you will choose from local files, then you can locate the Fastq files where you have stored your or saved your extracted Fastq files, and then just you have to select those Fastq files together, and then you can do the once you select the Fastq file here, then you will press on the start button, and then these data will start getting uploaded in your Galaxy web server. And once these are getting uploaded, they will come in the history panel. Okay, then these Fastq files will show up in the history panel. This means you have uploaded the data in the Galaxy web server. Okay, these are the Fastq files which you have uploaded, or these are the Fastq files are the raw data which you have uploaded, right? And it will show up in the Galaxy history panel, the right side panel. And once the uploading is done, then this gray color will become green in color. This means that this data can be used or for the further processing. Okay, so once we have all the data, what we can do is, once you have uploaded the data, what we can do is we can, with this pen symbol, we can edit the name of the file. Okay, so here these are the Fastq files, and below that there were the Fastq files which we have got from the raw data. So I have few files like this. One is wild type, rep1, then read1. Read1 is read pair 1 because it is paired ended data. Another is wild type, rep1, read2, because for all each of the replicates we have two reads. Then next is wild type, rep2, read1, right? Similarly we have done for file type and the treatment condition. Okay, so here once I have now I have to select for the quality control, because if you are doing the downstream analysis in any NGS dataset, then first you have to check or go through the quality control just to make sure that whether the dataset you are using for your downstream analysis, whether that dataset is it reliable or not. Okay, so how we are going to do this? We are going to do this with the tool that is the FastQC tool from the web-based Galaxy. Okay, so here I have used the Galaxy plant because I have already told you our dataset is the Arabidopsis dataset. So we are using the plant-based Galaxy web server, that is useplant.galaxy useplant galaxy.org. Okay, so here once I have logged into the database and uploaded my FastQ files to the history panel, then I'll go for the FastQC tool to check the quality control. Okay, once I'll search for the FastQC tool, then this panel will appear. Okay, and from here in the middle this button I'll press, and from here this is for multiple data set. Okay, from here I can select the multiple data set. So I'll select all the FastQC files which were uploaded earlier. Okay, it include all the wild type with 3 replicates and 2 reads. Okay, and all the treatment condition having 3 replicates and 2 reads. So all these files will be selected over here, and then there is a contaminant list. Contaminant list is the different primers, or if you want to deselect any of the region of the genome from here, then you can upload that the sequence file over here. And the adapter lists, which it's include if you want for the particular adapter, or you can leave it like nothing selected, then it will take it from the default as a Illumina adapter, it will look for it. Okay, and then there is you will go for execute, and once you put execute, then these files will appear over here. Once first they will gray in color and then they will get yellow in color. Yellow means they are processing, and once the data is done, then they will come in the green in color. This green in color means the data, these are the output of the fastq file. Then for each of the file you will see two data for of the fastq raw. One is web page, another is raw data. Now we will look for what is this, the what will be the output of this fastq data. So for this, what you can do is you can just press on this I button. This I button will make you view all the outputs of your fastq file. Okay, once you will press on this I button, you will get this file, and this file will show you the basic statistic or the summary of the fastq report. The fastq report means the quality check report, and this file will contain the basic statistic per sequence based quality, per sequence quality score, per sequence based content, and per sequence GC content, per base N content, the nucleotide content, and distribution, and then the sequence duplication level, over representative sequence, and the adapter content. So this data which we are getting, it is the first quality check on the raw data. Okay, this means their adapter sequences are also there in this raw data. Okay, right. So let us understand what is the basic statistic. So in the basic statistic you will get first the name of the file, which I have renamed to wild type rep1 read1. So this means this is the result, fastq result of the file of wild type rep1 and read1. Okay, so the file type is the conventional base calls. Okay, so this means this is giving the quality scores for the each of the bases here, and then this the platform here is Illumina, and this will gives the total number of sequences over here, and the sequence is flagged as poor quality. So means there are no sequence which is of poor quality, it means it's 0. So the minimum sequence length is here is 50, and the GC content is 44, which is normally in any plant genome, right? And here you will get the different graphs over here. So this graph is of per base sequence quality. So per base sequence quality means you can see that these these are the positions in read in base pairs. Okay, and you can see that these are showing the quality score across all the bases. Okay, so these scores are more than 20, right? I have already told you that when the Fred quality score is more than 20, then it is supposed to be a good quality data, right? And when it is 60, then the base call accuracy is 99.99%. Okay, so all of it is 20, above 20, so this means the base sequence quality is very good and the data quality is good. So the next is per when there is a, and you are seeing here that in some of the summary there is green tick, and in some of the summary there is cross. So green tick means that it's passed this quality report. Okay, and the sequence is well enough to further processing. So, and there is cross also. The cross means there's a warning sign, or it fails the QC test. So here the per base sequence content is showing the cross mark. So the per base sequence contents means the ATGC. In each of the bases, in each of the sequence, the amount of ATGC, when the amount of ATGC is not conserved, I mean it's not uniform, then it shows the cross sign, or it is less than the per base it is not equal, or all over the sequence it is not conserved, kind of like the amount of the ATGC it differs, it is getting differed. So Maybe the possible reason is that because right now we are doing the fast quality check in the raw data only. That's why the per base sequence content is not good enough, right? Once we remove all the adapters, okay, and all the adapters from both the end, then this per base sequence content will automatically get increased. Okay, so there is no need to worry about. So now with the adapter trimming, we are going to remove the adapters which we have ligated or the which were ligated during the sequencing, right? While the sequencing, I have told you that there is the fragments were being created after the fragmentation of the genomic DNA, and there is a ligation of the adapter. So it is important for the downstream analysis to remove these adapters before taking these reads further for the you know, the dataset analysis. So with the adapter removing, we will be doing using the tool Trimeomatic. Okay, so Trimeomatic, it is the tool which can be searched here. Okay, we will search for the tool Trimeomatic. Okay, then this will show the Trimeomatic tool giving the flexible read trimming tool for Illumina Ngs dataset, right? So when you will click on this tool, this central panel will appear here. In this central panel, here in the central panel, you can see that first you have to put the information about the single-ended or the pair-ended reads, whether your data is single-ended or pair-ended. So obviously we have selected the data for the pair end. Okay, so we will select the pair end here. Okay, and then the Fastq files, these are the input Fastq files for the first pair, that is read 1, and then the read 2. So here, while selecting the multiple dataset, we can select number of files of read 1. This is the wild type rep3 read 1. This third refers to the wild type rep2 read 1. And then fifth wild type rep1 read 1. And here along, we will select the treatment read 1 of the rep 1, rep 2, rep 3. Okay, then from the fastqc file read 2, we will select all the read 2 files of all the different replicates of wild type and treatment. Okay, then we will keep the setting in defaults, and there is one option, select the trimeomatic operation to perform. That will select for the sliding window trimming. Sliding window trimming means it will perform the trimming approach. Will start the scanning at the 5-5 prime end of the sequence and clips the read once it will reach the average quality within that window. Okay, within once the reads will fall below the threshold value, then there is some threshold value. Once the read when the reads are below that threshold value, then only it will clip from there that 3 prime end, right? So then we will click on the execute over there. Below here there is execute button, we will click on that, and from there we can see that there are the two files as generated. One is the Trimeomatic on rep3 read 2. One is paired file for read 2 and one is paired file for read 2, and another is unpaired file for read 1 and unpaired file for read 2. So here we can see that for each of the read, like read 1 or read 2, this Trimeomatic will give the unpaired file and one is paired file, right? So this unpaired file is means that this file has the mate of this file is not present or discarded from the read 2. That is why it is grouped into the unpaired file. Okay, so this means that after trimming with the Trimeomatic, some of the reads can be discarded from one file, right? After Trimeomatic, some of the reads are discarded from one file. Like if some reads are discarded from R1 file, then these reads are only present in the R2 files. Then we will group that reads into the unpaired file, because its pairing partner in the read 1 file is already get discarded while trimming process. Okay, so only a small fraction of the reads become unpaired and they can be ignored. So this is the result from the trigonometric, and from here what is the we will we will move ahead with the paired file only. Okay, then from here we will move ahead with the pair file only after this trimming process. After this trimming process, there is the alignment. So once you have all the reads, these reads, all the reads, and you have removed the adapter from all the reads, then you will align these reads with respect to your reference genome, right? So for that, what we have used is we have used the HiSet2 software, okay, or the HiSet2 tool from the Galaxy web server. So this HiSet2 tool will select, again you will select for this HiSet2 tool from this tool panel, and you will see the HiSet2, a fast and sensitive alignment program. This is basically we use for the alignment of the sequences with the reference genome. So here the source of genome will be the built-in genome because it is a plant web server of the Galaxy. So we will select the reference genome is the Arabidopsis thaliana, and tier 1 is a repository for the Arabidopsis thaliana, the database. Tier 10, sorry. So you will select the paired ended or single ended library. Our library is paired ended. Then again you will select the Fastq files. Okay, so here what you have to look for it now for the alignment file or the alignment process. Now the input of your HiSet2 will be the files from Trigonometric. Okay, now the files which we have got after the removal of adapters will be used as an input for HiSet2. So now here when we will select the multiple dataset, we will select the paired file from the file type and the treatment condition in each of the replicate. For both the condition, all the replicates. Okay, so we will select here we will select the read one file trigonometric, like this is the treated 12-hour condition and replicate read one. Okay, that is the paired file. Again we will select all the paired file, okay, from the file type and the treatment condition, and here we will select for the read to all the paired file of the read to from the file type and the treatment condition, and then we will go for the default setting and press execute, right? And when we will press execute, you will see the alignment file here having the mapping summary and the aligned reads. Here what happened is the HiSet2, this is the alignment software, it gives the BAM file, right? The binary alignment mapping file. BAM file we have used, it is the output of the HiSet2 and it is in a binary format. Okay, that is why the size of this file is very less, because it is in binary format and can be easily used or downloaded for further further process. Earlier there were different other alignment tools, or also they were used to give SAM files. The SAM file is of very sequence alignment mapping. The SAM file is of high size, or you can say the greater size as compared to the BAM files. So the SAM files are basically converted into BAM files, but here the HiSet2 gives the result as a BAM file only. Okay, so these BAM file will be used further for the processing. Okay, so now we will look for the mapping summary from this alignment using the HiSet2, and this is the output of the HiSet2, the mapping summary. Here you can see that the alignment rate, it is the 97.10%. It means your sequences in the genome has aligned properly. So what if your alignment rate is less than less than 20% or something like that, and if it is sometimes the condition happens like you have done the Arabidopsis uh, whatever the RNA seq, and what happened, your alignment rates comes very less or low. So there are the different reasons could be for that. One is that maybe your RNA quality is not good, that could be reason, or your the sequencing processing is not good, that could be reason. So what we do is, before going for the DE-seq analysis, we always do the analysis of the alignment to the reference genome. And if you are doing with some other crop which do not have the reference genome, or the genome is still not sequenced, then we will go for de novo assembly. But that is another topic, or another kind of RNA-seq analysis with the help of de novo assembly. So now till now the alignment is done. It will show you the result of like how many reads were there and how many times the reads were not aligned exactly one time. So basically the overall alignment rate here is 90% point 97.10%, 97.10%. So this means that the alignment rate is pretty good. So your sequences are aligning properly with your. So after the alignment, we will go for the counting. Counting means we'll go for the countings of basis. Like it will tell you about, like if there is some of the sequences are there, it will tell you about like how many times that particular genes is expressed in your cDNA. Okay, or you can say the expression. It will count the expression of that particular gene, okay, or the sequence, right? So for that, the one more thing you need to upload here, that is GTF file. GTF file is the annotation files that are present, okay. It gives you the information about the genome, okay. So GTF file for Arabidopsis, we can download the GTF or the GFF files. These two are the annotations files. We can download it from the Arabidopsis database, that is tier 10. Or if you are working in another crop plant or animal system, then you can also download their GTF annotation file from their databases, right? So here we will again search for the tool, that is HTC count. So this tool count the aligned read in a BAM file that overlap features in GFF or GTF file. Okay, so the input of this HTC count would be obviously the BAM files from the HiSet tool. So can you see here like what is happening here? If you are going with the workflow, then once you have the output of one tool, then this output is used as the input of the next tool, right? So that's why we cannot interchange the workflow. We have to go with the pipeline only. Okay, so here what we when we search for the HTC count, we will click on the HTC count. This central panel will appear, and here you will look for the data. Like here for the you will select the multiple data set, and you will here select the data like for wild type replicate 1, replicate 2, and replicate 3. There are 3 BAM files, and you'll select all of the same with the treatment files. Okay, and then we have to download the GFF file and upload this file again in the data set. Here I have downloaded the GFF file. Okay, it is there in 113 number, present over somewhere below here, okay. Then we will select the annotation file over here, okay. And then once you press the execute button here, then the count files or the count seq data will form in the will process in the history panel. And once it is processed, the files will get green in color, and you can see the result as follows. uh, as follows, like this. This is the result of the HTC2. This is the result of this file. I have highlighted this over here. Okay, so this is the result of the HTC count for the HiSet2 data on data 94 and 93. So data 94 and 93, that is this replicate 2 of wild type. So this means that this particular accession has this number has expressed this number of times in your cDNA. Okay, you can say. So this counts it counts the number of reads in the BAM file that overlap with the your GTF file. Okay. Now this HTC count files will be used as the input for your differentially expressed gene, or you can say in the DEC2 software which we are going to use for the analysis of the differentially expressed gene, right? So here now the identification of the differentially expressed gene using this tool DEC2. Okay, so this is the software is of utmost important which we are using to find out or the identification of our differentially expressed genes, or the genes which are getting higher or getting up regulated or down regulated as compared to your file type under solid stress condition. Okay, so here what you have to do is, here is the thing is, you have to first again search for the tool DEC2, then there the DEC2 tool will appear which determine the differentially expressed feature from the count tables which we have generated from the HTC2 count. Okay, so here what you have to select, here the different factors you can write down like control and treatment. Okay, so first we have to select here the treatment condition with respect to treatment condition because we are selecting the differential expression in treatment condition with respect to the control condition. Okay, so here in the treatment condition we will select all the HTC count files. Okay, HTC count files, this file type, sorry, HTC count files of the treatment condition rep1, rep2 and rep3, and in the second factor level we will write it down, name it as control, and we will select the HTC seq file HTC count files of the file type rep1, rep2 and rep3. And with the default settings we will put the execute button, and this will take some time. Okay, all right, all these things and all these tools will take some time to process these data. That's why I have not shown you the live session of this thing, because the generation of this data set or this output will take much time. Sometimes when the server is slow or the internet is slow, so it takes time to get updated over here, right? Once you have executed your DSeq2 data, then there is the plots are generated for the DSeq2, and there is a file are generated for the differentially expressed gene. So here this is the output of the DEC2 data, which is showing the plot. First is the PC plot, that is principal component analysis plot. It will show you the difference between it will show the variance or the difference between the your treatment and the control condition. Here you can see that your the all the replicates of the treatment condition control condition, the wild type falls in uh, here at a one place, whereas the treatment condition, here they are fall together. So this means that your data set is really of good, or the the variability within the replicates is very less, right? The variance is 97%. Okay, so and there are different data set or also outputs are also there. One is the dispersion model. It shows the dispersion about the same the wild type and the treatment condition. So the main thing that is of your use is the DEGs, okay, that will be generated. So this is the table which you will get after the DSeq output or the DSeq file. When you will click on this view button, you will get this data. Okay, when you this I button, and in this you will get all the DGs having the base mean value and the log 2 fold change value, with the standard error and their vault statistics, p-value and the adjusted p-value. Okay, so you can download this file from here. This is the save button. You can download this file having all the DEGs. Okay, and this file is in .csv file is this. Then you have to convert this file, and I'll show you how to convert this file also. Okay, so once you have all these CGs, and now you have to get through these accession numbers. What are these accession number belongs to? So for that we will do the annotation, right? Because we should know what these accession numbers are. So for that we will do the annotation and the extraction of your DSeq2 output. So we will search for DSeq2, and we will get another tool, that is annotate DSeq2 or DSeq output tables. So here you will select multiple select. If your data is in multiple set, you will select for the annotation annotated, sorry, you will select for the DSeq2 output tables. Okay, here I have selected the wrong data. You have to select for the DSeq2 result or the output tables, right? Which is this file, 138 number file. You have to select 138 number file over here, and then you will select the GTF format or the GTF file, the annotation file of for your genome, reference genome, and then you will press the execute, and then you will get the file with the additional column of the chromosome here. Here you Yes, here you will get the file with the additional number of columns which belongs to the chromosome, then start, chromosome start and chromosome end. Then there is plus or minus strand, and then there is where this gene is protein coding or RNA coding, and then the gene name. Okay, then again with the view data you can view this file, and when you press on this data, then you can download it with that similar button. Okay, with that save button or the download button. So you will have the CSV file here. So now you can download the CSV file from the DSeq2 data or the annotated DSeq2 data. So from now the CSV file we have to move it to TXT file, and then to the Excel sheet, data to the Excel sheet. Okay, so first what you have to do is you have to rename your CSV file to the .txt file, and then open a new Excel file. From here, option in toolbar, you can export the data in the tab delimited form from the data option, and then from text you can move your data to the delimited form using the data option, and then press next and then finish. Then with this your data will come up in the Excel sheet or a Excel form. I'll show you in the next slide, like this. Now your DGs would look like this only. In the first column you will have the gene IDs, then base mean, then log2 full gene value, standard error, wall statistics, p-value, adjusted p-values. And as I have extracted the genes from the annotated DSeq2 data, so there you can see the chromosome number, start and end of the chromosome, strand information, and then for protein coding, gene information, whether it is protein coding or RNA coding, and then followed by the gene name. Okay. Now from here, from the gene name, you can filter out the gene name, like the genes which are belonging to the NA, that are non-specified or the unannotated gene. Here you can filter out using the filter out option in the Excel. And what you have to do is the gene name, we don't want the gene name having the NA, the non-specified or the unannotated gene. So we'll put the filter of this not equal to NA. Then we'll press OK. Then what will it give? The result of the gene name which do not have the NA gene, right? So now we will have the gene name which does not belongs to NA. So here we have filtered out the gene column having the normal gene, having all the gene name except the NA, right? So here you can see that this after the gene name, this is the last number of genes which we have got, and here you can see that in the p-value there are many of the genes having the NA means. This means that whenever in the list of DGs, when you go downwards, then the number of significant DGs will decrease, right? They are the genes which they are the different DGs which has no expression values or a low amount of expression values. So we have to filter out all of these DGs and the genes on the basis of the p-value. We have to find the significant DGs which will be of any use to our data or to our result or our downstream process analysis. So we have to filter out the DGs on the basis of the p-value and then on the basis of the log2 fold change value. Okay. So here what I have done is, here we can uses the column only gene ID log2 fold change and adjusted p-value along with the gene name. Here you can then filter out again with the help of the filter tool in the Excel with the adjusted p-value up to 0.05, right? The significant where you will have the significant gene ID, or to being more stringent, you can look for if you are getting higher number of genes, then you can go for lesser or equal to the 0.01, then you will get the more stringent with your p-value. You can adjust according to your usage or your data, right? Then you will select the adjusted p-values having the less than 0.05 p-adjusted value, and then you have to filter out the DGs. Then you will once you have the significant p-values, you will go for the DGs. You'll select for the upregulated genes, you will select the genes having the log fold change greater than 0, right? Then all the upregulated genes you will get in the one side, and same with the downregulated gene, you have to select for the number of log2 fold change value having less than 0, right? Then you will have all the downregulated gene, and that's how you can fetch out all the DGs which are being upregulated and downregulated just using with the help of Galaxy Web Server and just Galaxy Web Server and Excel. Okay, so that's it for the hands-on of the Galaxy Web Server, and what I could see is that it is very beneficial for those who don't know the programming and nothing about the programming or the bioinformatics expertise, and they're still working on the this data. So Galaxy Web Server is just a very easy way to understand and to fetch out the
Thank you, Ms. Diksha, for a very well-spoken presentation. Welcome, Professor Ote. I can see you on the screen. So it was a wonderful presentation, and you had a lot of data, which you put in a very, very simple way. Though I know that for most of our first-year students, it sounded like French, but I'm sure this type of exposure was extremely essential at this stage so that they start learning the ABC of omics approach ultimately. So I'll throw the session open for any questions in case any student or faculty member, researcher wishes to ask Diksha about the technology. You can raise your hands, please. There are none in the chat box, if I'm not mistaken.
Oops. Oh.
Yeah, there is one.
That was mine only. Sorry.
Yours only.
I was looking for conversation, like questions in the chat box.
Okay. If there are no questions, we'll get back to Diksha through mail another time.
Yes,
because
sure.
most of our faculty is busy in taking classes and exams simultaneously, and therefore the right type of questions would have come from them rather than undergrads at this stage. So thank you so much, Diksha, for a wonderful presentation, for your time out of your research.
Thank you.
Okay. Best wishes and thank you. Look forward to seeing you again.
Yes, ma'am, sure. Okay, thank you.
Welcome. Now, I would like to take this opportunity to welcome Professor Ute, who is a renowned scientist and also happens to be the guide for our former student, Sabarna. Welcome, Sabarna. Before going over to Professor Ute's address, I would like to introduce her. Dr. Ute is a professor and head Department of Plant Cell Biology in University of Bonn, Germany. She has a long postdoctoral research experience from some of the renowned research laboratories across the globe and is presently the chair of plant cell biology in University of Bonn, Germany. For the last 21 years, she has supervised more than 20 PhD theses and almost double the number of bachelor and master students' theses. She is a recipient of a lot of funds to support her research work. She has a long list of publications in peer-reviewed journals. Dr. Ute is also on the panel of reviewers for journals and grant agencies. Specialized areas of her teaching interests are plant physiology, biochemistry, and cell biology. She is a proud recipient of Excellence in Teaching Award of the state of Bavaria. She has also organized advanced lecture courses on the origin and evolution of mitochondria and chloroplast and workshops on plant organelle signaling. With this very brief introduction, I would welcome Professor Ute and podium is yours, ma'am. Please.
Thank you very much for this nice introduction. And I would also really much like the organizing committee for inviting me to present a little bit about what we are doing. And before I do so, I would also like to mention one thing, and that is since over 10 years now, I'm in a panel to select DAD stipends for PhD students from India. So indeed, I have a long history with India, and before Corona stroke, I have been 8 times in Delhi each year for this selection, enjoying the hospitality and yeah, also seeing a little bit of your wonderful country. And we are also happy all the time to take up new DAD stipend students in our laboratory. However, today I would like to tell you a little bit about our research. And since this is a omics, is focused on omics, I thought that I'm not going to present to you detailed research, something that might not be so relevant for many of the students joining here, but would rather tell you a little bit how we use omic approaches, both in the research questions of our individual PhD students, as well as on the other side in a very large multinational EU-funded research consortium. So I will see, I think I should be able to share my screen. I'm not so experienced, here it is, with Teams. Can you all see my presentation?
Yes.
Very good. And I would like to have a pointer. Yeah. So since a couple of years now, I'm head of the plant cell biology in Bonn. And one of the questions that we really would like to address generally in our group is how plants respond to stresses. Because within their environment, there is a lot of changes in environment in form of abiotic stress, meaning things like drought, flooding, heat, and cold, but also biotic stress in the attack of pathogens and insects. And in most cases, plants have to respond to these stresses on an internal cellular level, because changing environment is a long-time adaptation of plants. But what we are talking about here is acclimation for a short time to a specific stress that either occurs very rapidly or only lasts for a few days maybe, or even only a few seconds or hours. So as you can already see here, there are specifically two types of stresses we in our group are concentrating on, and these are the abiotic stresses, drought and cold. But in this large EU project I'm also going to talk about, we are looking as well into heat and into flooding. So specifically, what we are interested in are the early stress responses. That means what happens after a stimuli has been received by a receptor and before any kind of changes on a cellular level are evoked. And one of the secondary messages that we have been studying for a long time now, about 15 years, has been calcium. Calcium is a ubiquitous volatile secondary messenger. In plants, it's involved in most, if not all, kind of stress responses. And it's so amazing that just the change in the internal concentration of an ion can then alter so many cellular processes. So by different calcium signatures, meaning length and repetition of the calcium signal, there are, and including a number of specific calcium sensors, there are very different processes that are altered, and gene expression, if we talk about here, transcriptomics is one of them, but it also directly affects metabolic enzymes. There is calcium-dependent phosphorylation, dephosphorylation, and calcium regulation of transport processes. And because I'm coming from the background of organelle, biogenesis, and function, we always had as one large focus the question how plant organelles are integrated into this calcium signaling pathway. However, calcium is just one of these second messengers that are responsible to convey these early stress responses. So a stimuli will create a calcium transient, but a stimuli will also affect other early responses. And so recently we have also started to look more into phytohormones, and they started with a project on jasmonate signaling. And since then, we are not specifically only looking at calcium on one hand, phytohormones on the other hand, ROS I should also mention here, but also how these different kind of messengers interact so that you have an overall response that is not just the result of one single of these messengers. And our start with phytohormones, as I said, was jasmonate signaling. And indeed, this was also our first start into transcriptomics. And I'm not going about any details about this. A lot of it is already published, but for me, this was a steep learning curve. And I think this is why I find a workshop like this so valuable, because one had to learn that it's easy to obtain the transcriptomic data, because it's easy to make the samples and you can easily buy now the Rna Seq. But the difficult part is then to curate properly this data. And we saw in the talk just beforehand a few of the things that have to be done before you can even start interpreting this data. But then you also have to be able to pull valid information out of these huge amounts of data that you normally get with an omic approach. And I would like to introduce now two student projects where we are using omics to try to tackle very specific research questions. And the thing with these student projects is, and also the ADAPT project, that we have gone one thing further. And that is our original research object has always been Arabidopsis, a model plant. The genome is known since the year 2000. So it is easy to get omics information and to interpret this omic information because the genome is so well annotated. However, in some newer projects, we have extended now our focus on crop plants, because if you think about what we want to do with all the questions of early stress responses is that we would improve plant yield up on certain stresses. And so we have extended now to two crops. One of them is barley in a nice collaboration with the University of Halle, and the other one in our ADAPT consortium is potato. And one of the, I would say, requirement for all of this kind of work that is met with these two crop plants is that indeed for both of them, there are reference genomes now fully sequenced and annotated so that data like your proteomics or transcriptomic data can even be analyzed and interpreted. So what are we doing with barley? So in barley, we are trying to see what happens when barley is exposed to H2O2. And H2O2 is an interesting thing because in plants, it is both. It is a stress indicator, for example, if something goes wrong with photosynthesis, but it's also used as a signaling molecule. So in case of H2O2, we have two kinds of responses. One response is the plant screaming something is wrong and I have to change, for example, gene expression, but it can also be a very deliberate signal, for example, for a biotic attack, and then it has to interact with other signaling pathways to get a specific response. And what we wanted to find out in this specific project is what is the calcium-dependent part of the transcriptional response to oxidative stress. So if we envision that oxidative stress leads to a change in gene expression, some of it might be a direct correlation, but in other cases, the H2O2 will evoke a calcium transient, and then there is calcium-dependent gene expression. And as you can see here, this was a collaboration with the University of Halle, Professor Edgar Peiter, and a postdoc in his lab. And in my group, it involved my long-year postdoc, Fatima Shikri, Zabana, who you will hear in after me, giving you some more input in the analysis of transcriptomic data, and Maha Giridan, who in her PhD project did the original characterization of a line that we used for this analysis and also provided the samples for the RNA Seq. What did we require to do so? And what we required was a line where we were able to define whether the H2O2 evoked calcium transient happens or does not happen. And so at the University of Halle, they created a barley line, a transgenic barley line of the variety Golden Promise that carries a calcium sensor protein apoequorine. This comes from the jellyfish Equora victoria, and what is less known is that this is indeed the protein that creates blue light, and by BRET, so direct transmission of this energy, feeds the GFP that we most of us know much better from this jellyfish. And the interesting thing about apoequorine is that if it is reconstituted to equorine, then this emission of blue light is a direct relation to the amount of calcium. So this can be used very nicely as an in vivo calcium sensor. And that you can see in this picture here. Here you can see the barley seed with the leaf and the root. And if you put a lot of calcium on it, then you can evoke a calcium-dependent calcium transient in the cytosol, which you can then read out as the blue light that is emitted. So what happens if you take such a line and you put it under stress? So here using the leaf tip and adding H2O2, you can see that you have a nice calcium transient. So the concentration of free cytosolic calcium increases very, very rapidly from 0.2 micromolar to over 0.6 micromolar, and then it declines, and in the long term, it will decline back to the basal level. And you can see that when we used this barley plant, we could see that the response was very similar to what had previously been observed in Arabidopsis. And then we added an inhibitor. So lanthanum chloride is a well-described inhibitor for channels, calcium channels that are localized in the plasma membrane. And the apoplast is one of the major resources for cytosolic calcium transients in plants. And you can neatly see here that where we have here this high transient up to 0.6, this here remains at about the basal level, which means that we have now a situation where we can induce a response to H2O2 in the one hand, but we can remove the calcium transient that comes with the H2O2 response. And what we did then is that we treated plants either only with H2O2 or with H2O2 and the inhibitor and did RNA seq. And this is where then Sabana came into play, who joined my lab a year before and turned out to be really good in doing all these transcriptomics analyses, so that he is now the one who is investigating these data. And what you can see here is first some data from the experiment without the inhibitor, so just what happens with the response to H2O2. And for example, this PCA plot just shows you about the quality of the data. So you can see we have 4 different sets of data. We have the root and we have the shoot, we have the control and we have the stress. And all our data sets are neatly separated. So that is a very nice show that the data are usable for our kind of interpretation. And indeed, it turned out that also other scientists have done similar experiments. There was a recent publication that did a similar approach, and we could see that our data and their match quite good. What they didn't do is anything about calcium, so that we expect that we will get a lot of novel information out of this approach. So once you have these data, you can then do all the analysis that are introduced to you in these workshops. So this you can see here is a cluster analysis, which identifies 3 different well-defined clusters. And there are obviously a lot of genes behind these clusters, and these have to be now detailed analyzed to get interpretation of these data, and you need subsequent further studies, because that is also something that was part of my steep learning curve, is that while it is nice to get a lot of these data, they don't mean much if you do not look afterwards, again, one-to-one, what does it actually mean for a certain set of genes or even individual genes. So what you can see is that if you look at the control versus the H2O2 treated plants, that about 1,100 differentially regulated genes can be identified with a certain cutoff. This was mentioned, I think, on this slide. You see it below here. So this is something that you have defined beforehand for you, where you think that the relevant cutoff is. And what you can see is about the same number of genes are up and down regulated. So we then looked what happens additionally if we add the lanthanum chloride to it, and thereby inhibiting the calcium part of the response. And what you can see here is that there are certain changes. And one of the things that surprised us is that if you look at the down-regulated genes, there was no much change. That seems to be reasonable if you think about that we are inhibiting a response. However, this is transcriptomics, and and this could also be negatively gene regulation, and that could also be inhibited. However, one has to be aware that these, while the numbers are very similar, are not completely identical genes, because there is a certain effect of lanthanum chloride by itself, so that indeed we have a separate data set with control plants treated with the inhibitor. But what is very clearly visible is here, when we look at the upregulation of the genes, that we have a reduction in number. And that is indeed, if you compare it, certain genes that are found in this set that are not any longer found in this set. And these are the genes we are now really interested in, because these are the genes where we believe that upon an H2O2 stimulus, they require a calcium transient in order to be deregulated, so that we can now have a look at those, and then can do one-to-one experiments to further prove that indeed these are calcium regulated genes. So this was Transcriptomics. This also is a proteomics workshop, so I would also like to very shortly point out that we are also trying to do similar experiments on proteome base, which is a little bit more elaborate. And because it is much easier to use Arabidopsis for this, or this is easier because the proteome of Arabidopsis under a lot of different conditions is already much better analyzed. So in this project, which is a collaboration with the University of Vienna, sorry, I didn't correct that here, is the work of another PhD student in my laboratory, Annelotte, and postdoc Andras, and will later on also involve curation of the data, where again Sabana might join the project then. And so what we did in this project is we wanted now to distinguish not what happens on the gene level, we wanted to see what happens on the protein level. So again, we used H2O2 and we wanted to know what kind of proteins are phosphorylated or dephosphorylated after the stimulus. And then again, we wanted to see how much of these is done by calcium-dependent phosphatases or kinases. And again, we used a line here, in this case an Arabidopsis line that carries this aquarine sensor. And you can see again here, similar to barley, that you have the response to H2O2 via a calcium transient. And again, you can see here that as well in Arabidopsis, this transient can be inhibited by lanthanum chloride. And then what we did is Annelotte prepared all these samples, and in Vienna we're going to do phosphoproteomics. And this will indeed give us two important informations, because we will see the change in proteome. This is something much less looked at so far generally. So there are a few papers out, but by far not so many as there is with transcriptomics. But it is important because we shouldn't forget that the response on a protein level is a combination of transcription, translation, and protein degradation. So one will see, and in some papers it has already been shown, that the alteration in the proteome are not one-to-one comparable with the changes in transcription. And in addition, because we are doing phosphoproteomics, we will get to know the changes in the phosphoproteome. So we will see which of these proteins require the stimulus H2O2 and calcium to be either phosphorylated or dephosphorylated. These were just some examples for individual projects in our laboratory. However, where the whole thing gets even more interesting maybe is what happens if you get a lot of groups together that can do a lot of omics. And there is more to omics than just proteomics and transcriptomics. There's also metabolomics and phenomics. And the project we are now working on since about a year is called ADAPT, Accelerated Development of Multi-Stress Tolerant Potato. And the question behind is how can we make potato fit for climate changes? So this is part of the EU Green Deal, where the EU would like to support research that addresses the question, what happens if our temperatures in Europe now rise to, in the worst case scenario, 3 degrees? Because there are certain numbers in place, and it is believed that EU, including the UK, there will be a bill of about 65 billion a year by this year, which is ahead in the future. But it's a large bill to be paid if we do not manage to either change this rise or adapt our plants better to the changes that we expect. So why potato? Potato is an important crop plant, not only in Europe. And potato is a cold climate crop. So it originated in the Andes, in certain regions in South America, and it has a peculiar characteristics that is sensitive to high temperatures during night. So it can rather well live under high temperatures during the day, but it doesn't like if it gets too warm in the night. And this is why even now there is a certain limitation. So in the north of Spain is a kind of a limit for how far south potato can grow in Europe. And as higher the temperatures are now getting, and even now this year in Germany, we had already night temperatures in May of 13 degrees or more, the growth of potato will be inhibited, or more specifically, tuber formation will be inhibited, and nobody wants a potato plant that doesn't have tubers. And so the aim of ADAPT is that we would like to enable breeders to develop varieties that are better acclimated and to do so faster. Because you all know breeding takes a long time, and if we can use molecular information, we can assist the breeders in finding the right varieties that they should cross. But in order to get this information, we have to find out how potato reacts and acclimates to these stresses. And there is much less known in potato than, for example, in Arabidopsis. And we would also want to know why already now some varieties are doing better than others, because this will identify the molecular targets we are looking for. And what you can see here in this picture is part of our consortium in a site visit at PSI in Brno last October. So who are we? And you can see that our whole consortium is kind of can be divided in 3 different subgroups. And one of them is a scientific community. These are universities or research institutions that already either worked on potato or, like us, worked on stress responses. And then, and that is very valuable, we have a breeding community. And indeed, these 4 breeders here represent not only the vast majority and most important breeders of potato in Europe, but are also one of the most important potato breeders worldwide. And they have the advantage not only to know a lot about potato, but they have the fields where experiments can be done under real conditions, and they have thousands of varieties that are so far not on the market and unexplored. And then we have a little group here, and this is a little group that will help us to increase the impact. Because in addition to assisting the breeders to select new varieties, we also do technology development, and here PSI is involved in improving high throughput screening of our varieties. We have with Europatad, the agency that supports the breeders and the farmers at the EU in Brussels. And here with the Argus, we have one of the agencies that allows new varieties in the market. So immediately everything that we get out here on our scientific level will be tested and also become relevant for the breeders, and here becomes relevant also directly for the farmers. And so this is a typical experiment that we are doing in this consortium. This was a pilot experiment done with a variety that we just choose as our kind of reference variety. And you can see that what we are doing here is we are exposing these plants under different stress conditions, both singular stresses and also combined stresses. And we do so in 2 forms. One is shown here, and this is in a high throughput facility. So under growth chamber conditions where machines can read a lot of phenomics information, like the plant size, you know, how the leaf looks like, the photosynthesis can measure it immediately, and so on. And we also take a lot of samples for further analysis. And in parallel, we are doing the same experiment on the field, which cannot then test so many of the different phenotypic characteristics, but we include drone flights that go over the field and also take samples. And indeed, it was very interesting to see that already last year, our whole idea, we wanted drought and heat stress had to be discarded because suddenly we had a year with a lot of rain and waterlogging became much more dominant. But it also shows how important it is to look for varieties that can withstand different of these stresses, because it is difficult to anticipate at the beginning of the year how the year will look like. However, I already mentioned we are taking samples, and here comes all the omics, because we are doing transcriptomics, we are doing proteomics, but in addition also metabolomics and hormonics. So we get an overall view on what changes inside the plant at a certain time of the stresses, and how does this affect something like the yield in tuber, but also how the plant in general looks like and grows. And so what comes out here are typical things like heat maps, or here is hormone analysis. So you get a lot of information. And as I mentioned before, this information is only valuable if you can curate and interpret it. And with the variety of data we get here, we really have another requirement, and we are really happy to have experts for that also in our consortium, and that is to use this multiple data set to then get information out of it, to make network constructions, to make dynamic models. So what we use here in this part of the project is that this group working on it fed all the existing knowledge into a database. They take all the information from these last screens, but also from individual experiments that the groups are doing. And then they use it to model the data to then come to certain outputs like regulatory hubs, phenotypic signatures. And for that, and this is a great thing about this consortium, they not maybe invent, but adapt tools for the visualization of the data, because somebody like me needs that to really get something out, because I can't interpret all these vast amount of different data by looking at the raw data. And so what you then get, as I already said, is certain things like connection properties, novel connections, regulatory hubs. But so what is what a group like me is doing there? Because I already mentioned, we are mainly concerned with early stress responses. And the disadvantage that we have in the moment is that what happens on that level, what happens with secondary messengers like ROS or calcium, can not easily be detected on a high throughput level. Indeed, very often we know very little about the dynamics of these things under a real nature condition. And so the part what we are doing here is that we are developing in potato sensor lines and gene reporter lines that will help us to address this question. So the sensor lines are lines, for example, that detect ROS, like the raw GFP, or calcium, like equarine, or indeed also gecko, because equarine cannot be used in high throughput screening conditions. Gene reporter lines means that we are identifying genes that respond specifically either to a certain factor like ROS or a certain hormone, or maybe even in response like something as drought. And then indeed there is again a reporter, for example, a GFP or some other reporter connected to the promoter of these genes. And these things can then be used to get an in vivo readout about what happens. And the advantage that we have here, because you could well say, okay, you can see what happens with all these genes if you look at your transcriptomic data, is that this can really be visualized while the plant is growing. So we selected these gene reporters and sensor lines in a way that they can be implemented into the high throughput facilities. So in the next round, when we do our next screening, we will not only be able to look at size of the plant, photosynthesis, and so on, but we will also expect that we get a direct readout what happens at least for some of these secondary messages and for some of these specific genes. here you can see a little bit how this looks like, right? You can see that under stress conditions, or in this case, it was not stress, it was as a test, the addition of the hormone jasmonate. You can see that you get a readout, and indeed, this is a readout that modern high throughput facilities can visualize. And so with this, I would like to finish. I hope I have given you a little bit of an idea how all these nice omics approaches can be used for different scientific questions. And a lot of people were involved here, only the ones involved in this research here. And yeah, also obviously a lot of funding is required in doing especially these studies as the high omics throughput multinational investigations like we do in the ADAPT. Thank you very much.
Thank you, Professor Utte. Any questions? I would like to throw the session open for question answers. So any questions from Professor Utte? Though I have some through the participants. So can I ask you?
Sure.
Thank you. Is there any scope of omics in industries?
What?
Scope of omics in industry.
Scope of omics?
Sorry, in industry. Yeah, I mean,
yes.
Probably what the participant wishes to ask, this study, can it be utilized in industry-based, production of something?
Not directly in
drug
production.
Designing or something like that?
Okay, drug design is a little bit difficult. I mean, you can use plants to do drug design, and indeed some of this can be used in order to optimize the system, because that is part, as I said, we are trying to optimize the system so that the high throughput facilities get better, because we can see molecular what is happening and compare to the phenotype that they can read, right? And that will definitely improve. And at the same time, if you envision you would like to really know when maybe a plant is making a drug, if you decide to go this direction, right? You could include sensor plants where your drug can be labeled, for example, or something else, so that you have an indication what really happens in the plant in real time, so that you, for example, might know the best time to harvest your plant, right? To then get to the drug, things like this. So yes, it's more indirect, but it can be utilized also in industry.
Thank you. The next question is, what is apoaquarin?
Oh, okay. So aquarin itself does not work per se. There is colenterazine. It's something that you have to add from the outside because the plant does not have that. And then apoaquarin and aquarin come together, and this changes the structure of aquarin in a way that it can take up then the calcium and create the blue light. So apoaquarin is the protein part of the aquarin sensor.
All right. Okay. Thank you. Does lithium-3 inhibitor exist in environment?
Sorry?
Soil, water, surroundings. This inhibitor that you mentioned, lanthanum chloride, is it available? I mean, is it found in soil, water, the natural surroundings of the plant?
Actually, I don't know, but not to my knowledge. So to my knowledge, this is not one of the things that is common in soil. It would also be really tragic for the plant because it's a really good inhibitor for the calcium channels in the plasma membrane, right? And this is required for most of the calcium responses. So no, to my knowledge,
no physiological relevance of this inhibitor then.
Not in outside the laboratory, yeah.
Yeah. Next question is, can it influence the plants in nature? More or less the same question.
It would if you dump a lot of it on the field, yes. So if you would flood a field with lanthanum chloride, your plants would be in trouble. We
There
see
is,
actually in case that interests, if you treat long-term with lanthanum chloride, we sometimes do like 12 hours beforehand, that you can see that this affects the internal calcium concentration in the cytosol. And that for a plant and for every organism is really dangerous because calcium, free calcium should be kept low in the cytosol.
What is RWC mentioned in pilot studies?
They are the real water content. So what we can also measure is we really know how much water is in the leaf at any time of our plant. That can easily be visualized.
And I would ask, I know you have a paucity of time. I'll ask you one more question, and that is by another participant. How can we get admission and masters in Bonn University? You have already generated interest.
Okay, we have a master program in plant sciences, and you just have to apply for it. And there is a selection panel. So each year we goes through, I mean, I'm not in the moment in this panel, through all the applications, and then we select about 60 students that get admission to this master course.
All right. Thank you so much. So I would like to propose vote of thanks and thank you for an extremely eloquently put up your research work through the presentation. Beautiful slides with a lot of clarity of thought. Also, today's talk was how simply she explained how to figure out research problem, and that too the one which is application based, like need to pick up potato as the study material, and also which property to study, which will be of use in times to come. I must tell the participants that Professor Ute is extremely busy, but still she consented to spare some time for this conference and also permit Sabarna Bhattacharya to give a session today. We are thankful to you, Professor Ute. I take this opportunity to express my gratitude to you on behalf of the organizing committee and Zakir Hussain Delhi College for your illustrious talk. Thank you once again, and we really look forward to have an offline lecture once you are in Delhi at any point of time. Thank you.
It is my pleasure. I would be very happy maybe in a year or so when I also can present detailed results from all our omics approaches, which might be much more interesting. It was really my pleasure. And as I could already see from the talk before me, I think your students will get a lot of really nice and valuable information about how omics work. And so I hope you will also then appreciate Sabana's presentation after me and have a very nice continuous project conference also, I think tomorrow and the day after.
Day after, 3 days. Thank you so much and have a wonderful day.
Thank you.
Now we have Sabarna Bhattacharya, and I request Dr. Sabinder to introduce Sabarna to the participants. In the meantime, I must tell the audience that Sabarna was a undergraduate student of BSc Honors Botany in 2018 to 21, no, 17 to 20.
15 to 18.
Let me check your CV.
15 to 18.
15 to 18, yeah, of course. Yeah, he was our student from 2015 to 2018. Our topper, and he has a lot of merit awards to his credit from university and from college. A very hardworking boy, and I recall he also participated in Science Congress, and he traveled with us to Imphal and presented poster there. And then he has an article on in Botanica also on what is the topic?
It's blue economy, ma'am.
Blue, yeah. What is that? Blue
economy.
Blue economy, right. So I'll officially now take the pleasure of introducing Sabarna Bhattacharya to the participants. Sabarna Bhattacharya had done his graduation from the Botany Department of Zakir Hussain Delhi College, University of Delhi in 2018. He completed his master's degree in plant sciences from University of Bonn in 2020. Sabarna joined the Wozniak lab, Professor Ute's lab, as a project assistant and a master thesis student in 2019, and has worked on hormone-mediated drought stress tolerance in plants. After joining as a PhD student in the same group in 2020, he has been employing relevant transcriptomic approaches to understand meticulous aspects of oxidative stress and drought stress signaling, not only in model plants, but also in relevant crop species in order to establish a comparative model of stress responses in plants. And I remember Sabarna also wrote an article and a chapter with me on jasmonic acid effects, a review article way back in, I think, 18 or so. So over to you, Sabarna. Welcome once again on behalf of the organizing committee and Zakir Hussain College, and over to you.
Thank you, ma'am, for those generous words of introduction. So before my video is played, as you know, everyone, that I have two segments, and I will be talking about the methods in RNA seq. So yeah.
Am I audible?
Yes.
Hello, Sabarna.
Hi, sir.
I don't know what happened to my laptop, and it stuck on its screen, and I was unable to do anything.
Okay.
So I'm not sure whether anyone have introduced Sabarna, or should I?
Yes,
Dr. Savinder, he has been introduced.
Oh, thank you, ma'am. Oh, welcome, welcome, Sabarna.
Thank you, sir.
So should I start playing the segment 1 of Sabarna's session?
Yes.
Yes, I think.
Yeah.
Sabarna, you wanted to say something before your video is played?
Yes, ma'am. So about the segments, which I already did, I have two segments, and basically just for everyone to know, I think I took the meaning of hands-on too much literally. So in this video, you will see me actually doing on a screen, which fortunately I could manage doing it, but later on I realized that it should have been more a pictorial kind rather than doing it. But I did it on a screen on a exemplary dummy data set. So maybe participants who have a laptop, if they have the proper software installed, they can do a little bit. So.
To be
Is it coming
with my
properly
college,
now?
once again after
Yes.
almost 4 years now, and it's indeed a real pleasure for me to talk in front of everybody, and maybe I was waiting for this day.
Sir, the sound is gone, I think.
Dr. Savinder, the sound is not there. I think you have unmuted yourself. You have muted yourself, sorry.
The first thing is
it coming now?
I will be doing everything
Audio is as
from
well?
my office here in Germany. So good afternoon, everyone. I am Sabarna Bhattacharya. I will be today talking about the basic methods of RNA sequencing in Linux and R environment. First of all, it's a pleasure for me to be with my college once again after almost 4 years now. And it's indeed a real pleasure for me to talk in front of everybody. And maybe I was waiting for this day to talk in front of everybody, and especially for this hands-on session. I am grateful to Ruchi ma'am and all the other organizers for giving me this opportunity to talk in front of everyone today. So before I start, there are a couple of things that I need to clarify. The first thing is I will be doing everything from my office here in Germany. So I have two computers actually. The first computer is based on Linux. That's the Ubuntu 20.04 LTS background. So the alignment of the transcriptome is normally done in this Linux environment. So my video is of two segments. It has two segments. The first segment is up to alignment, so where I will use the Linux computer. And the second segment will be an Windows environment where I will be using the R programming language. So just that it has two segments and it should not be a problem at the end. Just to let you know that. So with this, let us get started. So let's start. So this entire procedure, that after you extract your RNA, you want to sequence it, and then you want to kind of analyze it. This entire sequence, this entire process, it's a journey from once you have the raw reads, you want to get the gene expression estimates. So what is the expression of each gene and so on and so forth. And as you can see, then there are a couple of mathematical, statistical, and computational tools which you can apply to basically get an overview of the entire data. So a transcriptome data is sometimes referred to as a global transcriptome data. So it's very informative, full of information, and you just act as a vulture. You know, it just scavenge the desired information out of an ocean of information that you need. So basically, where it all started was in this information flow, which is generally referred to as the central dogma, where the genetic information flows from the DNA to the RNA, and which is then translated into the proteins. Because there are undergraduates here, I believed, I thought that maybe it will be worthwhile to start with something like this. So DNA goes to DNA, that's replication, goes to RNA, that's transcription, and RNA goes to proteins, that's translation. The DNA is essentially the double-stranded molecule, RNA is the single-stranded molecule, and protein becomes a complicated structure with many levels of helices, many levels of structure within it. So actually, the information on the genes of a particular organism, as you know, is contained in the mRNA, which later on gets translated to the proteins using potential machinery that is available. So we know that there are three types of RNA. As you can see here, the ribosomal RNA, the mRNA, which is, as a matter of fact, quite important for us because it contains the necessary information to get translated into the proteins. And of course, you have the tRNA. However, all these three RNA types play an important or significant role in the entire process in cell. So, and obviously in the eukaryotic mRNA, as you can see on the right hand side, it has the features of a cap, the poly A tail adapters, and so on. So what exactly is done after you extract your RNA and it goes to the company or a person who does the sequencing actually? So this sequencing has three ways of doing the sequencing. First is the Illumina, which is the very commonly used. As I said, I mean, it all depends on your target research, what is going to be the outcome, and accordingly you choose the required type of sequencing. So in this case, I think the Illumina is used in most of the RNA sequencing. There is also the Pacific Bioscience or PacBio reads, also the nanopore reads. Essentially, theoretically speaking, these all come up with sequence reads. However, there are a little bit differences between the various methods. So the Illumina, as you can see, the fluorescence intensity of the nucleotides is measured with colored chromophores, and by PacBio and Oxford Nanopore, obviously they are measuring the nucleotide individually, but it's in a different way. So essentially, as you can see in the figure C, we need to get the transcript out of it while analyzing it, and there are certain issues associated with it, like are the isoforms being considered, are the because everybody knows that there is the splicing phenomenon. So there is exon and intron, and there is the splicing phenomenon. So naturally, I mean, there is this concept of splice junction or exon intron junction, which nowadays can be included in a alignment procedure, and most of the aligners that are used for this purpose, they have special options to modulate all these features that are there normally. So basically, this RNA Seq analysis works like this. I mean, as I can reiterate once again that there are many kinds of RNA Seq depending on the type of molecule you want to sequence. I mean, there are standard RNA Seq. There is, the single cell RNA Seq, where you have the information from each cell and so on and so forth. However, every RNA Seq data that you get has to be aligned and has to go to further processes. So essentially, the downstream process that follows after your RNA is sequenced is similar in each kind of RNA that is sequenced. So first, what do we need to do? Once our sequencer, our company, has given the RNA to us, so we do a quick adapter check that the adapter has been ligated from both the ends. So especially here in Bonn, the company, the University Hospital Bonn, so they normally ligate the adapters and we get it. But however, it's better to give it a check whether the adapters are present or not, and if present, then there is a specific program which can help you to basically cut off the adapters, and then we do a quality check using a software called Fastq, and then we proceed to the alignment process. So the alignment process is twofold. The first one is can be a common one. I mean, that's alignment against a reference transcriptome. So commercially sequenced transcriptomes are available. So basically what you do is you take your raw reads and you align it against the reference transcriptome. The second, what is very interesting and very laborious, I think, at the end, it's de novo assembly. That means a commercial transcriptome is not available for the organism. So there comes the question of aligner change. Okay, so some aligners are meant for this alignment against a reference genome, whereas some aligners they proceed with a de novo assembly normally. Like aligners which are specific for de novo, I mean, which are used for de novo assembly, like Trinity, Salmon, Callisto, and so on and so forth. These are very specific for de novo assembly approaches. So after the alignment, so until the alignment, as I talked in the first slide, until the alignment, you can stay on Linux background, the standard Ubuntu shell, which is known as shell. That's the interface between you and the computer and the OS. Actually, that's the shell. There are different kinds of shell, Bone shell, C shell, and so on and so forth. But I will not go into those meticulous details. Just to keep in mind that until the alignment, it's done in Linux environment. It's better to do it in Linux environment. However, in the recent days, there is a very useful software which is known as Jupyter Notebook, where on a Windows background you can just run Linux and easily do it. I mean, people who don't take the risk of having to separate operating systems due to maybe hard disk usage or whatever, they can go with Jupyter Notebook. However, personally, I have never used Jupyter Notebook before, but I have heard that it's similar. I mean, it's like on a Windows environment, you can run Ubuntu shell and you can do the necessary analysis. So after the alignment, the important thing is count matrix. So we will be talking in the second segment. So count matrix is essentially you have all of your gene IDs together and you have the expression estimates, the count estimates. I mean, what is the count of every gene in that organism under those specified conditions. Then, of course, we'll go to some advanced, just a brief outlook into some advanced features like data visualization. So most common in RNA sequencing is the concept of heat maps. So, and then we go to advanced interpretation. We will see some machine learning approaches, some deep learning approaches, how to deal with your data once it has been successfully analyzed. So there are some important terminologies which needs to be actually learned before we move on to the actual hands-on doing of the alignment and so on. So there is a thing called thread score or pH read score. So that's the quality score of your RNA determination. So normally it should be around 30 to 40. I mean, in my previous transcriptome projects, it was, I think, around 32, 33, and so on and so forth. So that means, literally means that one base would be incorrectly inserted out of every 1,000 or 10,000 bases. That's the essential criteria. Then, of course, we have a terminology which is known as RNA integrity value or RIN value, so to say. It's basically the ratio of the distance. So as you can see, we have the eukaryotic RNA, and you can see these two bands, the 28S upper band and the 18S lower band upon gel electrophoresis. So there is a statistical way to calculate the ratio of the distances that is obtained. And normally the person who is doing the, who is expert in the sequencing part can help you with that. So that score is very important before it actually goes through the cDNA library processing and so on. There is also this term what is called as fragment. Fragment is essentially small piece of oligonucleotides, typically several hundred base pairs, subjected to an individual sequence determination, which is very important. Keep this in mind because we will go across this once we go to gene quantification in the second segment of my video. Next is also a very tricky term that is known as unpaired and paired end read, or single end read or paired end read. So basically, unpaired means sequence is reported from one end of the fragment, and whereas paired means sequence is recorded from the both ends of the fragment. And accordingly, you have to do some improvisations while aligning this to your reference genome, which is actually very important in this case. So as you can see in the top figure, it talks about the single end reads, and that's the reference sequence, and the second, the lower portion of the figure shows the paired end reads, which is the like both ends of a fragment are representing one single read. So that's essential, and that's also customizable because you can ask your sequencer to give you either unpaired reads or give you either paired reads. It sometimes depends on region also. Like Asian regions, mostly China, South Korea, they prefer paired end reads mostly. Whereas in Europe, I think it's more customizable, but in the Asian countries like most parts of China and South Korea, they go with paired end reads sequencing actually, and it's actually a lot of data. Because it's logical, right? So both ends carrying one information and one end carrying one information. So there comes the data volume actually. So mostly if you have paired end read data, so I think one replicate will be around close to 3 GBs of information. I think 3 or 4 GBs of information. Okay. Here comes a very interesting change, interesting parameter, which is calculated after you have aligned your sequences. So this is log FC, or logarithm of fold change. That means the expression change under two conditions. However, it can be sometimes a bit misleading. So let us say we have two genes. This is an exemplary data set, and we have its expression under stress conditions. Let us say it's 345, expression under control conditions. Let us say it's 49, and we calculate the fold change and the log fold change accordingly. So it's stress by control. That's always done. And accordingly, you have, if the log fold change value is plus, if it is a positive entity, it means it's upregulated, and if it is a negative entity, it means it's downregulated. And there are other things which is considered. I mean, we will discuss it in the second segment, but there is a term, this adjusted p-value. So basically you have, so this expression under stress is an average of the replicates, you know, because always an RNA-seq has to be performed using some certain replicates, certain number of replicates. So to say, the minimum should be 3 biological replicates per sample. I mean, you can go up to, I think, so normally I think I have seen going up to 6 replicates, 6 biological replicates, 1 sample, and so on and so forth. Accordingly, you take the average of the expression estimates under stress and control conditions, and you then calculate this logarithm fold change values. Point to be noted here that it's not always that the log fold change is the main criteria of your entire analysis. It always depends how you want to present your data after you have received the initial round of analysis. So many people, what they do is they just look at which genes are significant and which are not. Then they just plot a bar plot of the expression estimates, and they cross confirm with qRT-PCRs and so on and so forth. So the system requirements can be critical, because at least for the alignment part. So I have written down some requirements. So basically this computer where I operate the Linux, I I use normally 16 to 32 GB of RAM and 1 TB hard disk, or 2 TB also is fine. But the essential thing is it should have a higher random access memory, because that would increase your processing time. Alignments can be sometimes really time consuming depending on your aligner as well. I mean, and it also can depend on the complexity of the organism you are working with. So I also work with some crop species. So in crop species, and I use top hat 2 aligner, it generally takes 19 hours sometimes. So as I said, I mean, it depends on a lot of factors, but yeah, if you have a good random access memory computer, so then that will be nicer for you. Also, you need to have a good internet speed. You need to connect to a remote server, which is usually not here. So in this case, I use the Top Hat 2 server, which is remotely connected in some servers in United States. So maybe that's the time consuming part. And the R portion, the R coding can normally be done in a normal Windows computer with a 4 to 8 gigabytes of RAM. So that's fine actually. Okay, so all this said, so let's get started slowly. I have some dummy data sets which I will show, and you can see how it is actually done. So let us go to the quality check of the Fastq files of individual replicates of Arabidopsis thaliana under control and stress conditions, and we will go to the alignment after that. So I will stop sharing my screen. This screen is stopped now. Okay. Okay, so I hope you can see my Ubuntu shell screen. So this is the command line where most of the experiments are done. So let me give you a brief flavor of how this thing actually works. So there are certain commands for installing, for updating before you do any Rna Seq analysis. So the very first command is what I normally use is sudo apt update. Okay, that updates your versions of the programs that are already installed in your machine. So you press enter. Unfortunately, this is in German. Sorry for that. So I quickly type my password. Okay, so it's updating. Now it has updated. I then go to sudo apt upgrade. So these two steps are sometimes very useful before you actually start with the Rna Seq analysis. Okay, so it's now done. So what I do is, so now there is some interesting terminologies here. So if I write cd, that means you have to let the computer understand where your current directory is, the working directory where you have kept your files, where these guys lie. Okay. So what I do is so in this case I have my file in a directory called document, and from there I can check it. So let's write document here. It differs. It might differ according where you are keeping your files. So now the computer is recognizing your first parent directory as document. Now there is a next directory where my file is. So let's go to there. This is Rna Seq new. This is your this is my next directory where my files are. So there are a couple of things to discuss here as well. So the command that is used for Fastq, so that's the quality check. So absolutely, I mean, like if you come across terminologies like Fastq instead of faster, don't get worried. Fastq actually means quality report along with it. So I type Fastq and I press enter. Then this guy comes up and I go to file and I open it. So I normally will go to documents, then I go to Rna Seq new. There it is. Here is the sample file. You click OK. As I told, this is just a dummy file, just to see you, just to show you how things are actually done. It takes a bit of time. So you can see there are many scores here. Actually, I think nowadays the most aligners they have some quality checks in there as well. But many people, I mean, it depends on your data, as I told. I mean, if your data is very poor, so maybe you need to sit with your data. You look at which base point the N content or GC content is more or less, and then you can screen it off by hand. And yeah, so it has per base sequence quality, per tile sequence, average quality, and so on and so forth. So that was the first step. The second step, which I will normally show you, normally would like to show you, is something which is known as cut adapt. Although this sequence that I showed, it has been adapted trimmed already. So you write cut adapt, and then it comes up. So as you can see, it has specific options. So you have to literally type the sequence of the adapters, okay, here, and then you type the name of your output files, and then you proceed. As I said, if it is customizable, I think you can ask the person who is doing the sequence to help you do the ligation of the adapters. It can be done, or you can choose to do it yourself. So after you are done with this step, now comes that, so I will stop sharing my screen. Now it comes that where should you get your reference genome, right? Online, there are a lot of reference genomes that are available, but most commonly you can use Ensemble. In my case, I use Ensemble plants because I mostly work on Arabidopsis thaliana and other crop plant species. So basically, it's simple. So just a second. I go to Google Chrome, then so go to here, type Ensemble FTP. FTP means file transfer protocol. So where you can actually download your data sets. So I go to here, then I go to FTP download. Okay, so as you can see, there is this entire collection which you need. So essentially what you can get is, first and foremost, is the GTF file, which is very important because GTF actually means gene transfer format. It's basically an annotation file because once your alignment is successfully done, let us say, the program, whatever program you use, it needs to know which gene is what. Okay. Otherwise, if you don't supply this GTF file, the program will get confused and you will get ID1 expression, ID2 expression, ID3 expression, and so on. So in that case, then you have to blast each ID, and then you get to know the nature of your gene, which is a bit tedious because you can understand. I think the model plant Arabidopsis has almost 30,000 genes, and crop species, maybe it's like more like 40,000 or 50,000 and so on. So that becomes a bit tedious work to do, which can be skipped, I think, if you want. Directly download the GTF file and use it. Okay. So this also shows there are some other formats like VCF, whole database, gene sets, annotated gene bank, and FASTA. So you can take the FASTA files. Normally it appears as chromosome files. So what I do is, if you get individual chromosome files, so I can show once again. So there is a very interesting command which is known as cat. Cat means concatenate. So you have this command, and let us say you have chromosome 1.FASTA, you have chromosome 2.FASTA, you have chromosome 3.FASTA, you have chromosome 4.FASTA, you have chromosome 5.FASTA, and so on. Then you put a greater than symbol and you write the output name, output chromosome, sorry, or let us say output annotation .FASTA. Okay. So in this way, you can concatenate all the individual chromosome files yourself. Why I am saying this? Because the computer, the shell environment, it cannot read the direct downloaded files that you have downloaded, the chromosome files, as the FASTA files. You need to prepare what is called as index, and index differs based on the aligner. So let's give it a look how the index looks like. So if you are using the aligner top hat 2, this is the aligner, that is the top hat 2. So as you can see, it uses the index from another file which is known as bowtie index. Okay, you can prepare it by hand. I normally prepare it by hand, and this can also take some time. I have pre-prepared all that so that our further analysis should not be a problem because of the time crunch. So I am just showing you the commands of doing it. And I will also show what an absolute alignment should look like in a few moments. So basically it's somehow written like bowtie 2 index, sorry. Sorry, that has to be some problem. So bowtie is also like a program which is used to do alignments. Sorry. second. Turn my camera off. It's just there was this problem with the command, so it's botai to build, and you give, so you have to prepare your index from those concatenated chromosome files. So as you can see, there is this option how to do it, and it's very easy to do it. However, it might be a bit time consuming as well. So I have done it basically for this purpose from Arabidopsis reference genome, and like it differs from aligner to aligner. If I have HiSat2, sorry, so you have a difference. So as you can see, this is different. This is HT2 IDX, and this index building is also different. It differs from aligner to aligner. So now that we know what files are necessary, okay, for the actual alignment, so let's get started. So top hat 2, this command is guide annotation new dot gtf, and then we go to refsec. Sorry, I think in this case it is botai index. So botai index, and then I go to the name of your file. So in this case I have a dummy file, so that's sample file, that's Fastq. Point to be noted, this is very important. If you don't put it and you go on aligning your samples, you will be in a very tedious situation. Okay, so it's always better to, oh, maybe I didn't type it correctly, so sorry. This is annotation.gtf and botai index, and also you have the name of the file. In this case it's sample file.fastq, and then you press enter and something good will start. So as you can see, beginning top hat run version 2, and it has started. Okay, top hat is a very slow program, but once again, I mean, you have to base your alignment in every project that you do to one alignment. It's better always to go. Point to be noted that the first version of top hat is deprecated. That's not used anymore. So the developer of this program, Paul Trapnell, he got furious once in Twitter that people are still using top hat. I made top hat 2, so you can go with it without any hassle, and it just takes a bit of time. So especially when I do my crop species alignment, I normally what I do is I put all those comments and I go home because it takes overnight to complete, because as I told, it can take 19 to 25 or 27 hours sometimes, and if it is a parent read file, it can take some more time. So I will also show you the way how to do a paired end read. So in this case, in that case, it will be same. It will be top hat 2 g annotation dot gtf, sorry, gtf, and then you go with botai index, and then you go with so it would be something which is known as sample left read dot fastq and sample right read dot fastq, and essentially otherwise it's the same. And actually in top hat 2 the paired ends takes a lot of time. Top hat 2, as you can see in the just a second, or maybe I will type it for you once again. Top hat 2 has some specifications as well. These are some improvisations which you can do, but you can also do it very easily with the default parameters. Should not be a problem. And rest improvisation you might decide to do during the downstream, during the differential expression analysis, and so on and so forth. So with this, I basically come to an end of my segment 1, which was the Linux based thing. And I will now start my segment 2, which is the R based processing of your aligned data. So thank you, and please stay tuned for the segment 2.
Is it visible now?
It
Is it
was
visible and
and
audible?
it disappeared.
It's disappeared.
I think Dr. Ruchi also trying to play. Dr. Ruchi, first you can try.
Dr. Tabassum, is it visible, the screen, my screen?
Screen is visible.
Okay.
Oh,
Okay.
okay.
Then it must be okay. Dr.
So
Tabassum,
welcome back,
is it visible
everybody,
and audible now?
to the second
Yes, ma'am.
segment after the alignment was done successfully. And we will continue to do the later part of this presentation with R. As you can see already, there is a bit change in my background, so I have changed computers actually. So all right, so I will share that part where we were. So basically we finished quality check alignment, and now we are into the introduction to R. We will do count matrix generation as far as I can possibly fit into this time limit, because these all things can, in reality, this can take up to like almost few days to few weeks. So I will try to fit as much as I can. So I will share my R screen now. So this is the R Studio. Point to be noted, R is the name of the language. R Studio is the platform to do R, to do the coding actually. And the previous thing was basically Shell. Shell is mostly C-based language, C-based computing, but this one is a bit different from Shell. And this one, every function that we will use belongs to a package or a library. So first we need to check what is our working directory where R will keep the output of these files. So we have to press Control plus Enter. So it shows users documents. Perfect. So now we call the function that is responsible for feature counts, which basically gives an output with the gene ID and the counts in the various samples. So we call this function. Okay, so the function has been called. Now we try to basically list the BAM files. So BAM basically refers to the format of the file that is generated after alignment. Point to be noted here, there are two brothers, BAM and SAM. SAM is also there. So many aligners generate the output in form of SAM, which has to be later on converted to BAM, which is done in a software called SAM tools, once again in shell environment. But luckily, I use TopHat 2 for this talk, and TopHat 2 can produce alignments in the BAM format. So we have to list the files. Once again, as you can see, we have to give the GTF file as well for proper guidance of the feature counts function. So let's start. Okay, so as you can see, there are three files, three control files, three stress files of BAMs, which has already appeared. Also, there is the GTF file, which has already appeared. Now we run the feature counts function. Okay, so as you can see, it takes the BAM files, it's the GTF feature type, and we take the exon. So we run it. So it's running. So basically, it's taking the input of the files individually. It's quite fast, I should say. It takes 30 seconds for each file. We have 6 files with the BAM annotation. So 6 into 30, that's close to 2 minutes, I think. So. And you can see also in the percentage of the alignments, that should be the standard actually. Although me personally, I don't take into account these percentages as such, but well, it depends also at the end, some journals can ask about the percentages, how are the alignments, how did the reads look like, how did the alignment percentage look like, and so on and so forth. So as you can see, stress2.bam has the highest here, 90.6% successfully assigned alignments. So we write the table, but we will also have a feature which is known as length, because we need the length of the transcript for generating the expression estimates. As I mentioned before, expression estimates, there are many like FPKM, that's fragments per kilobase million, reads per kilobase million, and the universally accepted transcripts per million, which we'll do in a second. So we convert these results into a text file. Okay, show some error. We'll solve it. Yeah. So as you can see, counts.txt now is generated. It's the text file containing the counts of your thing. So what I will do is, let me show how it looks like. So basically what I will do is, I will take the txt file and I will put it into a Excel file, and then it will be better for the viewing. So I stop my screen sharing now. And so basically I take the counts file. All right, so it generally looks good. So I'll share my screen now. So as you can see here, very neatly the thing has come out. So now you can understand where we started. We started as a Fastq file. From there we traveled to the BAM file, and now we have this file. This file has clean gene ID, the length of the genes, and the counts for each gene. So just we make a bit of changes here, basically removing the .bam annotations, the .bam labeling. So this is now prepared. Now the very important thing comes here, that you need to do certain things before you move to the differential expression analysis. So I will separate out the gene ID part, the gene ID, the counts into a separate file, which I will later on put it as a file. So this is your although length has come here also, so, and I will put only the lengths in a separate file. Point to be noted here that I am always, when I am dealing with Excel and R doesn't basically read your file if this first thing is empty actually. So you have to make sure that it starts from right away here. That's A1, it should start from. Your labeling should start from here. Because if you leave this empty by any chance, you can face problems, because then it would be not a data frame, because this entire Excel sheet is read by R as a data frame. If you don't use this, then R will start reading it as a matrix. So that will be a different kind of a problem at the end. So where you are using an Excel sheet, you will use it as a data frame. So this should be marked then. So I have separated the length, so maybe I will delete it as well. I am sorry once again, because this entire thing is in German. So maybe I'll put it separately. Okay, so what I will do here is I will put this entire sheet into a separate Excel file. So let us say this file, and this file we will save it as, although I think maybe I don't know whether you will see my screen or not, but I am saving this as a separate file now. So this is, let us say, counts talk, so Excel sheet, and we'll save it in our working directory. So this is now saved. We will close it and we will go to the length part, and we will also try to save, we'll also try to save it as a so length file. And we'll save it in the documents. Length, let us say it's length doc. So now it's saved as well. Okay, so now we move to the differential expression. I will come to the TPM, that's the expression matrix, later on, because here it's a very significant thing and might be time consuming, because as you can see, I have to improvise at certain points, certain commands and stuff. So if we go to the screen now, so let us say, we start the screen sharing once again. Okay, so now we are at the differential expression analysis. Differential expression actually means that expression in stress with respect to control, or if you have let's say wild type and mutant, so it's in mutant with respect to the wild type. So that should be the way you should understand these things. So because we have to keep in mind, we should not reverse this way, because this is a common mistake which happens, because you neatly arrange the counts and at the end it becomes control versus stress instead of stress versus control. So this becomes a problem, and for that we need to do the transcription transcripts per million expression matrix, and we'll do it in a moment. So but first I want to show the differential expression analysis, because I think that some more improvisations in the command is required, because as I told you before, these all commands I wrote it quite a couple of months back, but as you see, small improvisations have to be done every time. So what we do is we first run this library dsec2, so it's not loading the entire library. Okay, so what we do is we take our counts data here. So we basically route counts doc, and these two guys, they are maintaining the matrix thing, because dsec2 normally reads matrix, it cannot read data frame, as I told you before. So these are some critical thing to be kept in mind. So I am reading the count stock file. Let us see. Okay, so it looks like I gave the count stock. Maybe I can take it directly here. So from Excel. Okay. Just a second. So here is our counts talk BAM. So let's save it, we import it here. Okay, so now it's imported. So we run this. We run this once again. Okay, so we need a metadata file as well. So what I should do, what I did already is I prepared a kind of a document for that, which has the sample information and replicate information. So we will import that as well. So we import this as well. So it's imported. It's M data. So I do this once again here. Data frame. We make another column for control and stress. So 3 replicates for control, 3 replicates for stress. Then we make it. Yeah. So it's now done. So now we go to the actual DSEQ results. So let's do it like this. So this is the first step of doing it. We are putting the count data, we are putting our metadata. Metadata, anything, anywhere you find the term metadata, that means anything which contains information on your data. So in this case, as you saw, we had 3 replicates of control. That's basically your metadata. So let us hopefully run this and let's see what happens. Okay, it's run nicely. So let us do this. Maybe I'll do. So it's these all things are very statistical, and we have to go through it because it's the way the differential expression and the significantly expressed differential genes are counted. So as you can see, it's fitting model and testing. I think as an apprentice to this very process, I find myself a bit, or let us say, I find this situation very magical. We started with just raw data, and with that you are coming up till this. So what we do is here we click on summary, press. So as you can see, the upregulated genes, LFC, and this is 1133, and the LFC less than 0 is 2014, 6.7%. And we have also filtered, as you can see, the low counts genes as well from this. But the condition that remains here is basically how to check which one was the control and which one was the stress. So for this case, what we do is here we write it as stress versus control. Second. stress versus control. So this portion basically filters your results and checks it whether it was done against stress versus control or not. It's very important to do it always. So okay, so now we are done. Let's take a look at the rest, which is our results. So this is your result actually. Now you should be, people who do this might be happy at some point, because here, as I told, I took a very raw data set which was never processed and the RNA quality was not good. Just an exemplary. So this P adjusted, that actually means the the P value without the false positives. So that means this P value is basically what is the raw P value. You cannot put an RNA seq result based on this raw P value. You always have to go with the P adjusted. That's what I follow. Now you can, once you take this entire data set, you can filter it according to, like, let us say this log fold change. You take a cutoff. Normally it is 0.5 or 1. So you can go with 1. I mean, if anything greater than or less than 1 or -1, you filter it out according to the P adjusted. So P adjusted is normally 0.05, or maybe stringent data set, you can take it with 0.01 as well. So accordingly, you can filter this out once you get this. So basically what we do here is we save it as a data frame. So df2, or let us say df3 data.frame, and we take data.frame this. Let us see what happens. Yeah, this df3 is done. It's a large data set. So this is how it is done normally. So as you can see here, the significance values are also here. I mean, it's, as I told you before, it's not a particularly very good data set. So I used it accordingly, but still you can find some significantly expressed genes like this gene 81G01490. It's significant and it's falling within our log fold cutoff. As I said, log fold cutoff can be set accordingly to your data. So that was with the differentially expressed data set. So basically log fold change means whatever is negative, it's down regulated. In stress, it is down regulated with respect to the control. Whatever is positive in stress, it is up regulated with respect to the control. So another interesting thing which can be done is here is to have the expression matrix, which is a bit tedious process, but I can show you how to do it. Yeah. So there are, so what I normally do is I design a function which can calculate it. There are many methods of estimating that. You can do it reads per kilobase million, and you can go with transcripts per million. Both are accepted. And so let's run it. So this function is ready. We do this read Excel, reading the files. Okay, so we have to be careful of our files, name of our files. So just a second. This is the length file. Length doc. Okay, so it seems like we did the reverse. Can happen sometimes. Okay, that has to be seen. So it seems like we have to modify our files a bit. Let us see. Okay, so let us try with this one that we don't have the gene ID. Maybe we save it later on, and because this order is never changed, so let us delete it. So we delete it and we keep only this. So I change the counts file, and I'll also change the length file. Maybe okay, length file is looking all right, but let us see what happens. So I import the count stock once again. So I imported the count stock counts file. So I do import. Yeah, it's now imported. Now let's see what happens. It's a bit changed now because I removed the gene ID thing. So let us see what happens. Okay, so it looks like that I have to change this thing. So there should not be any character in the length file. So I'll do it accordingly. Okay, so I think I did it. I did the lengths new file once again, and I saved it. Okay, so now we will import it. So this is the lengths new. Let us see what happens. Okay. Some more changes to it. As I said, a bit of improvisation is required sometimes in R. Let us see what happens now. So it's not recognizing it. That means I will put the entire thing once again. Something else. I apologize for this, but yeah, as young people who start to do this, these problems can happen sometimes. So take the length one. That is newly done now. So take the length one. So now the TPM is done. And in the same way, you can run the RPKM. As you can see, this can be a bit tedious with R, not only with R, but any programming language. This type of, on the point improvisation are sometimes needed. And especially we have, and this is a hands-on session, especially we have a lot of young people or young researchers who are starting with this bioinformatics. And especially people who do coding, I think it's always, they need to have some hands-on with this kind of improvisation. So if you look at the TPM file, so it's looks something like this, and now it's generated. So there is only 2 more steps left in our RNA sequencing for process for today. So let's do this. I need to shortly charge my camera, charge my laptop. So so now we are left with a few more parts. I will just go through a brief detail with them, which is the k-means clustering. The clustering, actually, the use of machine learning in heat map, and machine learning in RNA-seq, and let's take a look at how it looks like. So share my screen 1 second. Okay, so we took a look at all of them physically. Just a second. Maybe we can also take a quick look at the heat maps. How do they look like? But maybe at the end I will show you. So let's go to the machine learning part. So machine learning basically means the use of specialized or advanced algorithms to understand or interpret your data. So we have a clustering always. That's the first and foremost important. So once you have that huge list of differentially expressed genes, then we put that to the into the machine learning approaches. So there are 3 approaches: supervised learning, unsupervised learning, and reinforced learning. So clustering falls in the unsupervised learning, and basically we make some algorithms and make the computer show us how to do some predictions, how to do some modeling on it. I will basically not go into so much detail, but let me show you what it absolutely is done. So k-means clustering, so to say, it's like, you have different colored apples or fruits, and you have different bags, and you need to put them, you need to group them. So this grouping is obviously done by this method, which is k-means clustering. There are other approaches to clustering, as you can see, as you have probably seen, the hierarchical tree clustering, which is the basically it's the tree map, the tree which appears. But this is also widely accepted, which after you have completely done it, you get to put your genes into some groups based on certain similarities, which is predicted by the computer itself. And we have a process to do that. It's known as seal-out method or gap statistic method, where the computer predicts the number of clusters that should be used, and then you put your data into it, and you have your clusters at the end. So I will take just a few more minutes to show you the heat map. So very quickly. So let's go to the heat map. So the function that is used normally is known as the heat map function, p heat map function, so to say. Actually, so I do it like this, p heat map, and you call the functions like this. So it is installing the it's calling the libraries. So then you go to import dataset. I have a already made exemplary dataset on it. So you can see the dataset should be here. Yeah. So this is the gene. This is condition A, and this is condition B. This is exemplary. So that's randomly typing it. I generated just to show you how does this thing really work. So first of all, we have to prepare a matrix of it. So let's prepare a matrix. Exempt heat map. It's already here. So I call this variable. Then I go to exempt heat map gene ID. Then I go to row names. This is a function which basically removes a character vector, which is as A1, cell A1 of your Excel sheet. So I do this, and so let me quickly check. Okay, it's gene. So this is the function that calls or that converts it into a matrix. So it's ready. So now we just write DMKC here. And an exemplary heat map you can see. As you can see here, you can customize the color, you can, and it's based on the log4 change. So the data is based on a log4 change. So that's one of the methods to produce a heat map. I'm sure, I think one of our speakers today at the hands-on, she's going to show by Galaxy. Maybe you will get a more, you will get a different version of how to do the heat maps on a web server. So maybe she will talk about it. So that's all from the analysis part, and maybe that's why I called it as a journey from the raw reads to this portion of data representation, this type of data representation. Also, nevertheless, there is always, just a second, share the fresh screen. Okay, there is always skepticism in big data. Well, I found that this picture maybe explains in detail what skepticism actually means. So basically, these data, when they are generated, their reliability is sometimes put into question. How reliable these are, how statistically sound these are, how random these are, because these are all done by a programming language. So people always put this into question, especially people who have studied biology in the older years, maybe let's say in the 80s, 90s. That sometimes this kind of big data sometimes can be problematic. Even there are some journals, especially in MDPI, like MDPI plants and so on, where they put the review comments. There you can sometimes, I think a couple of months back, I saw the comment that how reliable is your data? We don't believe this data. So this type of comments, I mean, we cannot do anything. I mean, this is a process, and every process or every method has its advantages and disadvantages. Nevertheless, there are some processes to confirm some RNA-seq data. Obviously, you cannot decide one morning to prepare the RNA and do the qPCR of 30,000 genes. So every person will die in this planet then. But what is normally done, that you do the analysis, you successfully complete the analysis, then you frame a story for your paper, for your journal, desired journal, and then you pick out your candidate genes to do the qPCR and confirm it with the RNA-seq data. So I think I will stop at this point, and maybe I will, yes, it is finally over.
Thank you so much, Sabarna, for this such a comprehensive and detailed hands-on on the methodology that we can use for the RNA sequencing and its analyzing. First of all, I would like to ask some question on behalf of the participants. There are certain questions, if you would like to answer them. So first question is this method apply only for studying human RNA?
Not really. As I mentioned in one of the slides, I think this is a universal method. Every RNA that is generated at the end, if you want to analyze it, it has to go through these universal steps. There are certain differences, obviously, because that depends on the organism that you sequence. But universally, this method remains the same. It's always alignment, processing, differential expression, and visualization. And many people have different ways to do it. That's it.
Thank you, Sabarna. Another question is, what are the bases for alignment process?
Sorry, I didn't get it.
What are the bases for alignment process? Alignment.
Yeah, that's a good question actually, because as such, for maybe yeah. So the gene expression that we get at the end, it's out of the alignment, right? So many people say that good quality of RNA, good quality of reads sometimes matters in your alignment, whether you have a good alignment or a bad alignment. Many people are against this notion. So there is a paper which came out a few years back that says that once you are pre-processing your reads yourself, in order to have a good alignment, then you are creating data bias. Many people were saying like that. So, but yeah, basis is, yeah, you have a good quality of RNA, and then you proceed with a good alignment, and you should check the alignment percentage at the end. So that's it, I think.
Thank you, Shabana. There is one more question, that are these softwares free of cost? I mean, they are available for the public free of cost, or one has to purchase them?
No, they are free of cost. You don't have to purchase it. It also sometimes depends on the kind of actually, these softwares are not softwares. I prefer to call it algorithms. People who have designed it years back, but these are available online anytime. You just have to know the right way to install them. That's it.
Okay, so full versions are available online?
Yes, yes, yes.
Okay, thank you so much, Shabarna. And it was really a delight to, get all this information in such a great detail. And you started right from the quality check to alignment to all the algorithms that you can use. It was such a detailed and comprehensive hands-on session that I hope, and I'm sure that all participants must have imbibed it and they must be getting benefited from your session. I think there is one question from a
Diksha.
participants. Yeah,
I was
Diksha.
expecting one question actually from her.
So Diksha, can we enable the mic for Diksha? Mic and camera for Diksha. Okay, Diksha Besht has written the question that why you have used TOEFIC 2 aligner when there are better options available these days.
Yeah.
Maybe I said 2 or star.
Yeah. Okay, so I was expecting this question, Diksha. Actually, there is no answer to it, to be very honest. I mean, it doesn't depend old or new. It depends what you are getting at the end. And I decided to go with TOEFIC 2 because if you see the manual of TOEFIC 2, it's a splice aligner. Okay, there are better options, definitely. But it's a splice aligner, and so far I think I have got very successful results with it. I mean, obviously, when I have the time crunch, I will have to go with very fast aligners like this.
I think star is also a splice aware.
Exactly.
Like
so yeah, I know. Yeah.
And the time would be I think the time will be lesser time. In lesser time, star can do the same thing, right? So
Yeah, because actually for me, I think what really mattered at the end is I should get nicer results to reproduce at the end. So mostly it has been TOEFIC with me, and I really don't get bothered by the time it takes. But yes, I have tried alternatives, as you told. I have tried star as well. I have tried Trinity as well for sometimes. And yeah.
For de novo assembly, right?
Yes. De novo also I have done once. So I think, Diksha, I think the universal statement is there is nothing as good as bad, only until you are getting your results. So
yeah, that is
also. Maybe it's for availability at that time, what software is what program is available to you. Yeah. That also has.
Well, both
I guess that also has.
Both are like publicly available software.
Exactly. So it depends. As you know, that we use the proverb that whatever you get trusted with first, you carry on with it. But it doesn't mean that I will not use any other aligners. So it's just that I use that. So.
Okay, no problem.
Thank you, Diksha.
Thank you, Suvarna. That was a nice presentation.
Thank you.
So I would once again thank you, Shabarna, and I take this pride in once again saying that he's our own student, and it's really delight to see our own students doing such a great work in their respective fields. So it was great, Shabarna, to have you here, and we look forward to have more sessions, more talks with you in future.
Sure.
Thank you so much, Shabarna.
Thank you.
And with this, we have come to end for our first day 1, and I hope this day 1 was enriching for the participants. And tomorrow, day 2, that is on 26th May, we'll be starting at the talk will start at 10, but I would request the participants to log in by 9:50. And by this, we can call it for the day for today. So everyone have a good day. Thank you.